Hello everyone. Just checking my voice, please wait. Okay. Hi Preeta. Hi Krishanu. Hi Ashley. Hi Karmaveer. Hi Shruti. Hello everyone. Uh, Today's topic of discussion is amino acid chemistry, protein chemistry, and metabolism. This is long topic, very important topic. We'll discuss the each and every aspect of amino acid and protein chemistry and their metabolism. We'll discuss the important LAQ. We'll discuss the important clinical cases from this topic. We'll discuss the important short notes. We'll discuss the important aspects of amino acid chemistry, protein chemistry, and protein metabolism here. Okay, this is a sort of revision for your university examination or for your professional examination. Now, see, this is a very interesting topic, and there are lot of clinical cases in this topic, lot of clinical cases in this topic, and that are frequently asked in the university examination, and they are having the classical symptoms, key features are there in this clinical cases. So we'll discuss these clinical cases, and we'll discuss the important key features of these clinical cases. We'll discuss the important LAQ, that is long answer questions. We'll discuss the important SAQs. We'll focus on the important MCQs on the particular topic. So here from the प्रोटीन केमिस्ट्री में तीन important clinical cases है जो इलेक्ट्रोफोरेसिस से related है. One is multiple myeloma. There is characteristic M band in the multiple myeloma. Characteristic M band in the electrophoresis. Multiple myeloma, it is a plasma cell tumor in which there is multiple punched out lesion in the bones. And in protein electrophoresis, you will get sharp M band in the gamma globulin region because of monoclonal gammopathy. Only single types of immunoglobulins they are produced. So that gives classical M band in the electrophoresis in the multiple myeloma, and there is excretion of benzones proteins in the urine in the patient with multiple myeloma. These are the light chain proteins which are excreted and which gives classical heat test positive, which has property to coagulate, form the coagulum in between 40 to 60 degree centigrade. So this is multiple myeloma. Classical feature is the M band in the gamma globulin region in the electrophoresis. फिर से हम इसको डिस्कस करने वाले हैं. Then cirrhosis of liver. In the cirrhosis of liver, you will get decreased albumin, and beta gamma bridging may be there. Beta gamma bridging or increased gamma globulin will be there in the cirrhosis of liver. In the nephrotic syndrome, as other proteins they are lost in the urine in the nephrotic syndrome, and because of larger size of that macroglobulin, alpha 2 fraction, alpha 2 macroglobulin, it is retained. So there is raised alpha 2 globulins levels in the nephrotic syndrome and decreased albumin level in the electrophoresis. So these are the diseases which can be diagnosed on the electrophoresis: multiple myeloma, cirrhosis of liver, and nephrotic syndrome. It is overlapping topic with electrophoresis also. Plasma proteins, electrophoresis, these are the overlapping topic. In both this topic you may get these cases. Okay, now this in the metabolism, these are the important cases. First one is the phenylketonuria. All of you know that phenylketonuria, it is a very common condition, inborn error of metabolism, autosomal recessive condition in which there is mousy order of urine. We'll discuss the detail again. Mousy order of urine, and it is confirmed by the tandem mass spectrometry. Mousy order of urine, hypopigmented patches, mental retardation, these are the features in the phenylketonuria. Alkaptonuria, black urine on standing. Black colored urine on standing and ochronosis means arthritis due to deposition of pigment alkaptone in the joints. Alkaptonuria. Then maple syrup disease, urine disease, there is smell of burnt sugar, maple syrup like smell of urine because of excretion of branch chain keto acids in the urine. maple syrup urine disease related with branch chain amino acid metabolism. Then homocystinuria, there is ectopia lentis. Ectopia lentis, that is known as subluxation of lens. There is myopia, ectopia lentis, long stature, tall stature, and the homocystinuria, there is classical subluxation of lens, that is known as ectopia lentis in homocystinuria, and cyanide nitroprusside test will be positive here. So it is ectopia lentis, means subluxation of ocular lens, and cyanide nitroprusside test positive for homocysteine. Nitroprusside test will be positive here. So various urea cycle disorders, they are characterized by the increased ammonia level. So we'll discuss one by one urea cycle disorders. Albinism, there is generalized hypopigmentation. Generalized hypopigmentation. It is characterized by the hypopigmentation. Also the also there is that is nystagmus. Nystagmus will be there, and photophobia will be there in the albinism. In primary hyperoxaluria, there is tendency to form the urinary stones, oxalate stones. Oxalate stones. So these are the common clinical condition: phenylketonuria, alkaptonuria, maple syrup urine disease, homocystinuria, urea cycle disorders, 5 urea cycle disorder, and one syndrome, that is hyperornithinemia. Then albinism, uh, then primary hyperoxaluria related with the glycine metabolism. Then Parkinson's disease, there is decreased level of dopamine. Carcinoid syndrome, there is tryptophan, more tryptophan is converted into serotonin, that causes the symptoms in the carcinoid syndrome. Then Hartnupp's disease, it is characterized by decreased absorption of tryptophan from the intestine, and it is characterized by ataxia, mental retardation, and the pellagra like symptoms because of decreased conversion of tryptophan into niacin, and pheochromocytoma, adrenal gland tumor, because of and it causes the increased synthesis of catecholamines. So there is increased blood pressure, there is headache. These are the symptoms of pheochromocytoma: increased BP, headache, and the blood pressure, headache, and the heart rate. Pheochromocytoma, adrenal gland tumor, and there is excretion of vanillyl mandelic acid in the urine. So these are the typical features of these clinical cases which helps in the diagnosis. ये जो features अपने दिमाग में रहेंगे तो हमको diagnosis करने में easy होगा. So these are the important clinical cases. Note down these clinical cases. These are very important. These are very important for the university examination, or you may get this PKU on this topic. You may get essay type LAQ. कुछ यूनिवर्सिटीज में ऐसे टाइप LAQs आते हैं. दिस इज वेरी इंपॉर्टेंट. phenylketonuria, alkaptonuria, homocystinuria, because they are related with one important pathway. So long answer can be asked on this topic. Also these are important SAQs and the viva questions. Important SAQs and viva questions. These are the important list of important LAQs from this topic. Just wait. Hello everyone. Good evening all of you. See, these are the important LAQs long answer question. The first one is structural organization of protein, that is primary, secondary, tertiary, and quaternary organization of protein, which is based on number of amino acid, sequence of amino acid, then folding of the polypeptide upon itself, assembly of protein into 3-dimensional structure, and it also depends on number of polypeptide chain. So structural organization of protein: primary, secondary, tertiary, and quaternary structure of protein. Second important LAQ is ammonia metabolism. How ammonia is formed in our body, means sources for the ammonia. Then how it is transported in our body, means formation of ammonia, transport of ammonia, toxicity of ammonia, and how it is disposed of by the urea cycle, how ammonia is converted into urea, and various urea cycle disorders. This is important, very important, the top rated question from the biochemistry, that is ammonia metabolism and urea cycle. Third important question is phenylalanine and tyrosine metabolism. तो कोई भी अमाइनो एसिड का जब हम मेटाबॉलिज्म लिखते हैं तो सबसे पहले उसका एक फ्लो चार्ट बनाना है हमको कि ये अमाइनो एसिड जैसे फेनिलएलानिन है, फेनिलएलानिन इज कन्वर्टेड इनटू टायरोसिन। सो टायरोसिन आपको क्या देगा, पहले ग्लूकोज देगा कि that fat dega. So depending on that you will start your answer. Just make a flow chart, like tyrosine will give you glucose, tyrosine will give you fat, and tyrosine will give you different specialized product. For example, tyrosine will give you melanin in skin, hair and eyes. Tyrosine will give you dopamine in the central nervous system. Tyrosine will give you the norepinephrine and epinephrine in the adrenal medulla, and tyrosine will give you T3 and T4 थायराइड हार्मोन्स इन द थायराइड ग्लैंड। सो पहले आपको ओवरव्यू लिखना है और उसके ओवरव्यू में और एक लिखना है ऑल अमाइनो एसिड्स दे आर इनकॉर्पोरेटेड इनटू प्रोटीन। सो टायरोसिन इज इंपॉर्टेंट फॉर द सिंथेसिस ऑफ ग्लूकोज, सिंथेसिस ऑफ फैट, सिंथेसिस ऑफ प्रोटीन्स, एंड टायरोसिन विल गिव दिस स्पेशलाइज प्रोडक्ट। यहाँ से आपको चालू करना है। और कोई भी मेटाबॉलिज्म जब हम लिखते हैं सो पहले उसका डिस्क्रिप्शन हमको लिखना है। डायरेक्ट पाथवे स्टार्ट करने के पहले आपको लिखना है कि व्हाट इज फेनिलएलानिन एंड टायरोसिन। सो दीज आर द एरोमेटिक अमाइनो एसिड्स, दिस बिलोंग्स टू नॉन पोलर अमाइनो एसिड्स, and this phenylalanine is essential. It is phenylalanine is essential and tyrosine is non-essential amino acid. So tyrosine contain phenolic hydroxyl group. So like that, just in short write down the description of that amino acid, try a particular diagram indicating that this amino acid will give this products. Okay, so यहाँ से हमको स्टार्ट करना है। सो दिस इज अगेन वेरी वेरी इंपॉर्टेंट क्वेश्चन, दैट इज फेनिलएलानिन टायरोसिन मेटाबॉलिज्म along with inborn errors of this metabolism, phenylalanine and tyrosine metabolism. This is important here. Then glycine metabolism is important. Then metabolism of sulfur containing amino acid, that is cysteine and methionine, is important. And tryptophan metabolism with disorders. This is favorite question for the NEET PG entrance examination. Also for the various entrance examination, for example FMG exam, typical, that is the tryptophan is responsible for the niacin synthesis. So deficiency of niacin causes the pellagra, that is diarrhea, dementia and dermatitis. So it is related with the niacin. So this is overlapping topic with the vitamin. So it is frequently asked in the NEET PG entrance examination. Also it is important topic for FMG and INICET exam. So these are the important LAQs from this topic. Then SAQs, short answer question. It includes the functional classification of proteins. Functional, I am talking about the functional proteins. See here, अमाइनो एसिड क्लासिफिकेशन इज डिफरेंट। अमाइनो एसिड का क्लासिफिकेशन अलग है, प्रोटीन का क्लासिफिकेशन अलग है। अमाइनो एसिड्स दे आर क्लासिफाइड ऑन द बेसिस ऑफ 4 प्रॉपर्टीज। फर्स्ट, their structure, second, their solubility, third one is the whether they are essential in diet or not, and fourth, metabolic fate. This is amino acid classification. And protein classification, protein, they are classified on the basis of their functions, also on the basis of their chemical nature and solubility. They are classified as simple protein, conjugated protein and the derived protein. Simple, conjugated, derived. But here what is important in the classification of protein, functional classification of proteins, means indirectly you have to write the functions of proteins. So how they are classified on the basis of their function. For example, catalytic proteins, it includes the enzymes. Transport protein, albumin, transport protein, like that. So it is functional classification of protein, which is important as an important short note. Then amino acid classification. Then biologically important peptides. Peptides matlab less than 10 amino acids. So peptides, that includes vasopressin, oxytocin, glutathione, then bradykinin, that is thyrotropin releasing hormone, gastrin, bacitracin, gramicidin. These are the examples of biologically important peptides. So this is important short note. Then plasma proteins with various disorders, just हमने discuss किए, multiple myeloma, cirrhosis of liver, nephrotic syndrome. Immunoglobulin structure and their functions, it is important, very very important for MCQs. Immunoglobulins with their important functions. And alpha helix or secondary structure of protein, you may get SAQ short answer question on the alpha helix beta pleated sheet, you may get the short notes. So these are the important short notes from the protein chemistry. And these are the important short notes from the metabolism, that is transamination reaction. Very very important short note, transamination reaction, transmethylation reaction, biogenic amines and their uses, specialized product of tyrosine, glycine, tryptophan, phenylketonuria, branch chain amino acid metabolism, and homocystinuria. These are the important short answer questions. So this is overall important topics from this protein chemistry or amino acid and protein chemistry and metabolism. Now we'll start with the topic that is chemistry of amino acid and the proteins. In that we'll discuss the important topics. We'll discuss the amino acid classification. We'll discuss the functional classification of protein. We'll discuss the biologically important peptide. We'll discuss the plasma protein disorders. We'll discuss the immunoglobulin, and we'll discuss the structural classification of protein, that is primary, secondary, tertiary, and quaternary structure of protein. These are the important topics from the chemistry part, and rest of the topic they are from the metabolism. So see, amino acids. So amino acids, they are classified on the basis of four properties. How many? On the basis of four properties. कौन सी four properties? They are classified on the basis of four properties. First one is based on their structure, based on their distinctive R group, based on their structure, based on their distinctive R group. They are classified on the basis of their structure based on their distinctive R group. Now see this is amino acid. See, this is amino group, this is carbon, this is carboxyl group, this is hydrogen, and this is R group. So depending on the distinctive R group difference in their structure, they are classified as aliphatic amino acid, aliphatic simple, aliphatic branch chain. They are classified as aromatic amino acid, sulfur containing, hydroxyl group containing, aromatic amino acid, amino acid. Like that, they are classified based on their distinctive R group. Okay. Second classification, it is based on the polarity. Polarity means whether they are soluble in water or not. It is based on polarity. Okay, if you remember the classification, if you remember it on the basis of their class, you can easily remember the polar non-polar amino acids. So this is second. Third one is it is based on the essentiality, whether required in the diet or not, means essentiality, whether they are required in the diet or not. And fourth one is the metabolic classification. Fourth one is the metabolic classification based on their metabolic fat. So this is how अमाइनो एसिड्स आर क्लासिफाइड. चार तरीके से इसका क्लासिफिकेशन होता है. पहला based on their structure, दूसरा based on the solubility in water, whether they are polar or non-polar, third whether they are required to be taken in the diet or not, means essentiality, and fourth one is the based on the metabolic fat. These are the four important classification, four important basis on which amino acids are classified. Now see, this is based on their structure. They are aliphatic with aliphatic side chain, that is simple and branched, then aromatic amino acids, acidic amino acid, basic amino acids, hydroxyl group containing amino acids, then amino acid, and sulfur containing amino acid, So this is classification based on distinctive R group. So aliphatic simple, aliphatic branched. See, in this structure, if in place of R, ये R के जगह पे आपने H डाल दिया, तो this becomes glycine. Glycine and alanine, these are the CH3. यहाँ पे CH3 डाल दिया, तो it is alanine. Branched, it is valine, isoleucine, leucine. So glycine alanine simple, branched valine isoleucine VIL, valine isoleucine leucine. Aromatic amino acid, all of you know the aromatic amino acid, that is phenylalanine, tyrosine, tryptophan and histidine. Phenylalanine, tyrosine, tryptophan and histidine. Acidic amino acid with their amides, so aspartic acid, glutamic acid means glutamate, aspargine, their amide, aspargine, glutamine, aspargine, glutamine. Then basic amino acid, his basics are loose. This is the mnemonic to remember. Basic amino acid, histidine, basics are loose, arginine and lysine. Okay, his basics are loose, histidine, arginine, lysine. OH group containing tyrosine, serine, threonine, threonine. So OH group containing Ti-Saris thread, Ti-Saris thread. This is mnemonic to remember the OH group containing. Amino acid, it is proline. It is proline. And sulfur containing, it includes the cysteine and methionine. Cysteine and methionine. So 20 amino acids, 21 is the selenocysteine, 22nd is the pyrrolysine. 21 is the selenocysteine, 22nd is the pyrrolysine. They are classified as, on the basis of distinctive R group, they are classified as, or on the basis of their structure, they are classified as aliphatic side chain, aromatic amino acids, acidic amino acid with their amides, basic amino acid, OH group containing amino acid, amino acid and sulfur containing amino acid. So this is classification based on their structure. Now based on the polarity, just remember the non-polar amino acids. How will you remember the non-polar amino acid? See, non-polar amino acids. Rest of the amino acids, they are polar. So how to remember the non-polar amino acid here? See, aromatic amino acids, aromatic amino acid, that is phenylalanine, tyrosine and tryptophan. They are non-polar. Phenylalanine, tyrosine, tryptophan, non-polar amino acid. Then the branch chain, branch, that is valine isoleucine leucine. Branch chain amino acid, they are non-polar. Then from the simple amino acid, it is branch chain, and simple amino acid alanine. Alanine is non-polar, and from the sulfur containing amino acid, it is methionine. Methionine, which is non-polar. So phenylalanine, tyrosine, tryptophan, valine isoleucine, leucine, alanine and methionine, these are the non-polar amino acid. The rest of the amino acids, they belongs to polar. Of these amino acid, in this non-polar classification, tyrosine is relatively polar among the non-polar amino acid. इसमें से ये सब non-polar है, लेकिन ये tyrosine थोड़ा सा soluble है, so it is relatively polar among the non-polar. वैसा ही सेम इसमें ग्लाइसिन में है. ग्लाइसिन बिलोंग्स टू पोलर अमाइनो एसिड, बट इट इज रिलेटिवली नॉन पोलर अमॉन्ग द पोलर. कौन सा? ग्लाइसिन. Okay, so these are the non-polar amino acid. Now all of you know essential amino acid, and this is famous viva question. Essential amino acids, how will you remember the essential amino acid? There is mnemonic, that is essential amino acid. So famous mnemonic, that is mat will fly. Mat will fly. So essential amino acid, 10 amino acids, they are essential, and this histidine and arginine, they are conditionally essential. They are required during the period of growth. They are required during the period of growth. So mat will fly. So here trick to remember is, this is mnemonic to remember the essential amino acid. But remember this this A is for the arginine, not for the alanine, because alanine is produced from the pyruvate by the transamination reaction. So A is for arginine, and all of you know that tyrosine is synthesized from the phenylalanine. So tyrosine is non-essential. So remaining two, that is threonine and tryptophan. They are essential amino acid. Tyrosine is not non-essential amino acid. So methionine, arginine, threonine, tryptophan, valine, isoleucine, leucine, phenylalanine. This is phenylalanine, histidine, lysine. These are the essential amino acid. Rest of the amino acids, they are non-essential. Of these, this arginine, and the histidine, they are conditionally essential. They are required during the period of growth. Otherwise, they are not required. So conditionally essential. So 10 amino acids, they are essential amino acids. Okay. And the next classification is based on metabolic fate. So it is very easy to remember based on their metabolic fate. See, glucogenic, based on their metabolic fate. And this is ये आपको याद रखना पड़ेगा क्योंकि इसके बेसिस पे हम LAQs लिखेंगे. एरोमेटिक अमाइनो एसिड, मेटाबॉलिज्म ऑफ ग्लाइसिन, मेटाबॉलिज्म ऑफ सल्फर कंटेनिंग अमाइनो एसिड, इसके बेसिस पे हम लिखेंगे और ये MCQ में भी इंपॉर्टेंट है. सो रिमेंबर, बेस्ड ऑन द मेटाबॉलिक फेट दे आर ग्लूकोजेनिक. Glucogenic means they will give glucose. They are known as glucogenic amino acids. Then both glucogenic and ketogenic. Glucogenic and ketogenic means give both glucose and fat in their metabolism. And third one is only ketogenic amino acids. Ketogenic amino acids. So it is easy to remember the ketogenic amino acids. It include leucine and lysine. Remember that if though both these are in the option, remember leucine is exclusively ketogenic. अगर दोनों option में दिया है, go with the leucine. Leucine is exclusively ketogenic amino acid. So these are the exclusive ketogenic amino acid that will give the fat, will give fat in their metabolism, will give fat producing amino acids. Both glucogenic and ketogenic, it includes phenylalanine, means aromatic के तीनों, phenylalanine, tyrosine, tryptophan and isoleucine. They will give both glucose and fat. They will give both glucose and fat, and these will all remaining all will be included in the glucogenic amino acid. So in this way, try to remember the amino acid classification. हमने क्या क्या डिस्कस किया अमाइनो एसिड क्लासिफिकेशन. ये शॉर्ट नोट है, ये वाइवा के लिए इंपॉर्टेंट क्वेश्चन है. वाइवा के लिए, ग्रैंड वाइवा जो आपका पेपर के समय यूनिवर्सिटी एग्जाम के समय में होगा, उसके लिए इंपॉर्टेंट है. ओके, तो हमने इसमें क्या क्या डिस्कस किया. सी, हमने डिस्कस किया कि अमाइनो एसिड्स दे आर क्लासिफाइड ऑन द बेसिस ऑफ 4 प्रॉपर्टीज. फर्स्ट वन इज द डिस्टिंक्टिव आर ग्रुप, मीन्स उनका स्ट्रक्चर. दूसरा पोलर है कि नॉन पोलर है. तीसरा इसेंशियल है. इसेंशियल है मतलब अपने बॉडी में सिंथेसिस नहीं होता है, वो बाहर से लेना पड़ता है, उसको बोलते हैं इसेंशियल अमाइनो एसिड. सेम इज फॉर द फैटी एसिड. इसेंशियल फैटी एसिड्स हमारे बॉडी में नहीं बनते, वो हमको बाहर से लेने पड़ते हैं, उसको बोलते हैं इसेंशियल. And the based on metabolic fat, whether they give glucose, they give fat, or they give both glucose and fat, they are classified. Okay, so based on the distinctive R group, they are aliphatic, aromatic, acidic, basic, OH group containing amino acid and sulfur containing amino acids. Polar, non-polar classification is there. Remember the non-polar amino acid, it includes aromatic, branch chain, simple amino acid, that is alanine, and sulfur containing methionine belongs to non-polar amino acid. So tyrosine is non-polar, but it is relatively polar compared to the other amino acids. Same is for the glycine. Glycine belongs to polar amino acid, and it is relatively non-polar among the polar amino acids. On the basis of essentiality, they are essential and non-essential. The 10 amino acids are there. They are essential amino acid. Mat will fly. A for arginine, T for threonine, T for tryptophan. There is no alanine, as alanine is produced from the pyruvate by the transamination reaction. There is no tyrosine because tyrosine is produced from the phenylalanine. Okay, so methionine, arginine, threonine, tryptophan, valine, isoleucine, leucine, phenylalanine, histidine and lysine, these are the essential amino acids. Rest of the amino acids, they are non-essential amino acids. And based on the metabolic fate, just remember this is very, very important for the MCQ. Exclusively ketogenic amino acid is the leucine. If leucine is not there in the option, go with the lysine. If both are in the option, go with the leucine. Which of the following is the exclusively ketogenic amino acid? मतलब it is leucine. अगर दोनों है दिए हुए तो leucine के साथ जाओ. अगर leucine नहीं है तो lysine आंसर रहेगा. lysine नहीं है तो leucine मतलब लाइसिन और ल्यूसिन दोनों रहा तो भी ल्यूसिन आंसर रहेगा. ल्यूसिन इज एक्सक्लूसिवली कीटोजेनिक अमाइनो एसिड. These are the aromatic 3 and isoleucine will give both glucose and the fat, and rest of the amino acid they are glucogenic amino acid. This is all about the classification of amino acid. Now see, glycine, alanine, sympon, valine, leucine, isoleucine branched, OH group containing serine, threonine, also tyrosine contain phenolic hydroxyl group, sulfur containing cysteine and methionine, amide group containing that is asparagine and glutamine, dicarboxylic that is aspartic acid and glutamic acid. Okay, it is having the one extra carboxyl group. So aspartic acid, glutamic acid, the dibasic amino acid lysine, arginine, lysine, arginine. Also histidine is the basic amino acid. Now see, glycine is the simplest amino acid. Glycine, it is the simplest amino acid. It is optically inactive, smallest, and it is important for the collagen synthesis. Collagen having the 30% of the glycine. Alanine, it is important. It is the most common amino acid which gives you glucose in the gluconeogenesis. Alanine, most common amino acid which gives you glucose in the gluconeogenesis. Valine, leucine, isoleucine. Leucine is exclusively ketogenic amino acid. Ketogenic amino acid, leucine. Serine, most of the enzymes in their active site they contain serine, so they are known as serine proteases. They are known as serine proteases. Hydroxyl group containing serine. Serine, it is the most common amino acid at an active site of an enzyme molecule. Okay. Then methionine. Methionine is the starting amino acid in the peptide synthesis. When there is synthesis of polypeptide in the translation process, methionine is the first amino acid which is coded by the AUG. It is coded by the AUG codon. Okay. Then amide group containing asparagine and glutamine. Now see this glutamine, it is a transport form of ammonia. Transport form of ammonia from brain and intestine. Glutamine, transport form of ammonia from brain and intestine, and from muscle it is transported in the form of alanine. Okay, aspartic acid, glutamate, all amino acids, all amino group from the different amino acids, they funneled or channeled into glutamate. So glutamate is the acceptor, central acceptor of amino group, glutamate. So lysine, arginine, these are the basic amino acids. Now polar and non-polar, these are the examples of polar and non-polar amino acids. So non-polar, see these are the non-polar amino acid. Then forget about this. So there are 4 amino acids that contain aromatic ring, that is histidine, phenylalanine, tyrosine and tryptophan. And these are the special characteristics of amino acid. Histidine, it is imidazole group of the histidine, it is important, and histidine, it is important in the buffering action, it is important in the buffer. Histidine, it is important in the buffer mechanism. Phenylalanine contain benzene ring, tyrosine contain phenolic OH group, it gives the Milan's test positive, phenolic OH group. Tryptophan contain indole ring, it is bulky amino acid, it is bulky amino acid tryptophan. And arginine contain guanidinium group. So these are the special groups which are present in the amino acid. So this is all about the classification of proteins. This is all about the classification of amino acid. Now moving to the second SAQ, short answer question, that is functional classification of protein. Now proteins, they are classified as simple, conjugated and derived proteins, but there is also one more classification that is known as functional classification of protein, and which is important. Just wait. हाय शेखर भाई, लिखना तो इंग्लिश में ही है. समझने के लिए हिंदी ठीक है, लेकिन लिखना तो इंग्लिश में ही है ना. This is for the revision of first MBBS students which are giving the exam in August. exam going students. Okay, now functional classification of protein. So मैंने पहले ही बोला proteins they are classified into simple proteins, conjugated protein and derived protein on the basis of their chemical nature and solubility. Their proteins are also classified as the complete protein, incomplete protein and poor protein depending upon the content of essential amino acid. मतलब 10 के 10 amino acid अगर उसमें है तो उसको बोलते हैं complete protein. जैसे एक प्रोटीन, मिल्क केजिन, these are the complete proteins. इनकम्प्लीट कब बोलते हैं? जब एक अमाइनो एसिड लैकिंग है तो उसको बोलते हैं इनकम्प्लीट प्रोटीन. जैसे पल्सेस में पल्सेस लैक्स मिथ्योनिन, सीरियल लैक्स लाइसिन, so it is known as incomplete protein. And the poor protein which lacks two or more than two essential amino acids, they are known as the poor protein. For example, Zane protein, it is lacking the more than two amino acids, that's why it is known as the poor protein. But here the important classification is the functional classification of protein. Functional classification of protein, it includes, the first one is contractile. Just wait. First one is the structural classification based on their structure, based on their structural proteins, based on their structure. So structural proteins, it includes collagen, it includes keratin, it includes keratin. All of you know that keratin is important in the hair and nails, skin, hair and nails. Collagen, it is important in various tissues, it is important in the bone, it is important in the cartilage, it is important in the basement membrane. So in the various tissue, collagen is important, it is a part of extracellular matrix. So this is important, structural proteins, collagen, keratin. Then second important is the storage protein, storage proteins. Storage proteins, it includes the, that is apoferritin or apotransferrin, it includes myoglobin, myoglobin, it includes apoferritin, ferritin. So these are the storage proteins. Myoglobin is important for the storage of oxygen, and it is it supplies the oxygen where during the period of muscle contraction. So it is important during the sternus exercise and muscle contraction. It is the immediate source. It is immediate source of the supply, will supply the oxygen. Myoglobin, apoferritin, storage proteins. Then contractile protein. Third one is the contractile proteins. Contractile proteins for the contraction. So actin, myosin, actin, myosin. These are the contractile proteins. Then catalytic proteins, enzymes, catalytic proteins, enzymes, catalysis that catalyzes the various reaction, enzymes, for example, lactate dehydrogenase, then uh, lactate dehydrogenase, alkaline phosphatase, or any superoxide dismutase. All these enzymes, all enzymes, मतलब जितने भी सब प्रोटीन्स है एक्सेप्ट राइबोजाइम्स, राइबोजाइम्स छोड़ दिए तो सारे कैटालिटिक proteins है. All enzymes are protein except ribozymes. So catalytic proteins. Then the hormones, that is regulatory proteins, they are known as hormones. Regulatory proteins, for example, hormones, various hormones, peptide hormones, they belongs to regulatory proteins. Regulatory proteins, hormones, various peptide hormones. Then the receptor proteins, so receptors, so receptors for hormones, so they are included in the receptor proteins. So structural, storage, contractile, catalytic, regulatory protein, receptor protein. Then defense protein, then transport proteins, defense protein, transport protein. And the one more is there, defense protein, it includes the immunoglobulins, immunoglobulins. Transport protein, it includes albumin globulins, albumin, it includes albumin, transport protein. And one more is there, defense, transport, and one more is there, so that is genetic proteins, genetic genetic protein. So it includes histones. So this is how they are classified, functional classification of protein. तो इसको आपको आप ये जो पार्ट है उसका एग्जांपल्स जो है ये इलेबोरेट कर सकते हैं अगर उसका क्लासिफिकेशन आया तो. सो रिमेंबर दैट प्रोटीन्स दे आर क्लासिफाइड ऑन द बेसिस ऑफ देयर फंक्शन. It proteins are classified on the basis of chemical nature and solubility, that is simple protein, conjugated protein and derived protein. Simple means only protein is there, conjugated means the additional prosthetic group will be there, and the derived means after the of the uh, first and second group the proteins derived, they are included derived proteins जैसे कि पेप्टोन्स हो गया, पेप्टाइड्स हो गए, these are the derived proteins. Okay. And the simple में globular proteins and fibrous proteins, so albumin, globulin, histone, protamines, prolamins, they are included in the globular proteins. And the fibrous protein, they include the collagen, keratin, elastin, these are the fibrous proteins. Okay. So simple, conjugated and derived, this is one more classification of protein. This is functional classification of protein, and one more is on the basis of nutritional basis, that is complete protein, incomplete protein and poor protein. This is how proteins are classified. This is how proteins are classified. Now coming to the important LAQ, discussion of important LAQ, long answer question, that is structural organization of proteins. And proteins are organized. So structural organization of proteins, there are 4 levels of organization of proteins. 4 levels. How many levels are there? 4 levels: primary, secondary, tertiary and quaternary. Primary, secondary, tertiary and quaternary. This is very very important LAQ for university examination. Primary, secondary, tertiary and quaternary structure of protein. Now primary, secondary, tertiary and quaternary, if there is single polypeptide chain, it has 3 levels of organization: primary, secondary and tertiary. If there is more than 2 peptide chain, 2 or more than 2 peptide chain, it is quaternary structure of protein. So depending upon, see, depending upon, so number of amino acids and sequence of amino acid, number and sequence of amino acid. Then second, folding of the polypeptide chain. Then assembly into 3D structure, assembly into 3D structure, and number of polypeptides, number of polypeptides, peptides, they are classified into 4 levels of organization, that is primary, secondary, tertiary and quaternary structure of protein. अभी मैं आपका एक हॉस्टल का एग्जांपल देता, देखो हॉस्टल जो है, द पूरा का पूरा हॉस्टल अगर आपने कंसीडर किया, सो हॉस्टल इज ए क्वार्टरनरी स्ट्रक्चर, और उसमें आपकी एक जो रूम है, एक जो रूम आपको दी गई है, दैट इज टर्शियरी स्ट्रक्चर, ए 3 डायमेंशनल स्ट्रक्चर विच इज ए फंक्शनल प्रोटीन. वो रूम जो है वो फंक्शनल है, वो रूम जो है वो फंक्शनल है, सो दैट इज टर्शियरी स्ट्रक्चर ऑफ प्रोटीन. पूरा का पूरा हॉस्टल की बिल्डिंग है, दैट इज क्वार्टरनरी स्ट्रक्चर ऑफ प्रोटीन. ओके. और डोमेन का मतलब क्या होता है, जैसे रूममेट्स है, मतलब एक आधा पोर्शन एक पार्टनर का रहता है और दूसरा पोर्शन दूसरे पार्टनर का रहता, तो वो जनरली वो उसी के एरिया में लिमिटेड रहता, वो इधर ज़्यादा ओवरलैपिंग करता नहीं, इधर ओवरलैपिंग हो गई तो झगड़े हो जाते. तो वो जो पार्ट है उसका वो डोमेन है उसका, और इधर का पार्ट है वो इसका डोमेन है. सो दैट इज डोमेन. गॉट इट. सो primary structure, secondary structure, tertiary structure and quaternary structure. So it is the linear, it is a sequence of amino acid in the primary structure, which is important. Number of amino acid and sequence of amino acid, it is important in the primary structure. The further coiling of primary structure will form the secondary structure, coiling of the primary structure will form the secondary structure. Further folding into 3-dimensional structure, it is known as the tertiary structure. It is known as tertiary structure. When there is 2 or more than 2 polypeptide chain, it is known as quaternary structure. 2 or more than 2 polypeptide chain is there. It is known as quaternary structure. It is known as quaternary structure. So primary, where number of amino acid and sequence of amino acid is important. Holding of primary polypeptide chain upon itself causes the formation of secondary structure. Assembly of this polypeptide into three-dimensional arrangement and the formation of functional protein is known as three-dimensional structure. It is known as the tertiary structure of protein, and when it is more than two polypeptide units, it is known as quaternary structure of protein. This is their how they are classified. So this is primary structure, primary structure. This is secondary. This is tertiary structure, and this is quaternary structure. Primary, secondary, tertiary, and quaternary structure of protein. Now see this. So how they are numbered and why the sequence is important here. So number of amino acids, they are important in the primary structure. Also the sequence of amino acid, it is important in the primary structure. Now see, suppose this is glycine, alanine, serine. This is one peptide, glycine, alanine, serine, and suppose alanine, glycine and serine. That's why the sequence is very important. That results in the formation of two different peptide. If the sequence change, it results in the formation of two different peptide. So in the primary structure of protein, we'll discuss the primary structure. Just wait. See here. Just wait. Primary structure. In the primary structure, the number of amino acids, they are important, and the sequence of amino acid is important. Sequence of amino acid, it is important. There is this primary structure, it is stabilized by the covalent linkages, and it is not broken down by the denaturation process. In the denaturation process, all other linkages, they are broken down except except the primary structure. It means except the covalent linkage, means peptide linkages, they are not broken down in the denaturation process. So here peptide bond is important. Formation of peptide bond is important in the primary structure. Now peptide bond, how it is formed? How it is formed? Now see, this is first amino acid. This is first amino acid. This is R1 COOH. Now this is second amino acid. This is R2. This is carboxyl group, amino group, carboxyl group. Now there is on dehydration, there is formation of CONH linkage. Now see this NH2C R1, this is H, this is CO. Then it combines, it forms the, on the dehydration, it forms CONH linkage, which is known as the peptide linkage or peptide bond. So this bond is known as peptide bond. Now what are the characteristic of peptide bond? Remember this, TRUP. Remember the characteristic of peptide bonds. In the primary structure में आपको ये लिखना है, number of amino acid, sequence of amino acid, peptide bonds. So bonds which are responsible for stabilization of primary structure is the covalent bond, peptide bond, and occasional disulfide linkage. They are also important in the primary structure. Now they are of trans configuration, they are rigid, and no rotation is allowed. No rotation is allowed around the peptide bond. So trans configuration, they are rigid and no rotation is allowed. Then they are uncharged. This is uncharged, and it is polar. It is polar and partial double bond in character. Partial double bond in character. So these are the characteristics of peptide bond. Trans configuration, rigid, no rotation is allowed, uncharged, polar and partial double bond in character. So rotation is not allowed around the peptide bond, but it can be allowed around the phi and psi angle. So at this angle, the rotation can be allowed, but at this peptide linkage rotation is not allowed. So it is number of amino acid, sequence of amino acid. It is important in the primary structure. Peptide bond is important in the primary structure. So how they are named, how this see. So this is a structure of pentapeptide having the 5 amino acids. Penta means 5 amino acid, not the peptide linkage. डाइपेप्टाइड में एक ही पेप्टाइड रहेगा, 2 अमाइनो एसिड रहेगी. ट्राइपेप्टाइड में 3 अमाइनो एसिड रहेगी, 2 पेप्टाइड लिंकेज रहेंगे. टेट्रापेप्टाइड में 4 अमाइनो एसिड रहेंगे. पेंटापेप्टाइड में 5 अमाइनो एसिड रहेंगे. सो हियर 1, 2, 3, 4 and 5 amino acids will be there, and 4 peptide linkage, 1, 2, 3 and 4 peptide linkages will be there in the pentapeptide. They are named from the amino terminal to carboxyl terminal. जब synthesis होता है peptide chain का, it is also from amino terminal to the carboxyl terminal. So this is pentapeptide. This is pentapeptide. For example, glutathione. If you take example of glutathione, it is gamma glutamyl cystenyl glycine. It is glutamyl cystenyl glycine. मतलब glutamate, cysteine और glycine, they are important in the structure of glutathione. So structural organization of protein, primary structure. So primary structure, number of amino acid, sequence of amino acid, it is important in the primary structure. Insulin, take example of insulin. Just wait, यहाँ पे है क्या देखने दो मुझे. See, insulin. When insulin, it is polypeptide. So now see, this is structure of insulin. Just wait. So it is see. This is insulin when it is produced. Insulin when it is produced. So insulin having the A chain and B chain. It is having the A chain and B chain. So suppose this is A chain. This is A chain with 21 amino acids in the A chain, and B chain 30 amino acids in the B chain, 30 amino acids. So A chain start with the glycine, B chain starts with the phenylalanine. A chain starts with the glycine, B chain starts with the phenylalanine, and this is connecting peptide. Suppose this is up to this, and this part is known as C peptide. This is connecting peptide, C peptide. What is importance of this C peptide? This indicates the endogenous production of insulin. मतलब अभी डायबिटीज में अगर आप डायबिटीज पेशेंट जो इंसुलिन थेरेपी पे है उसका आप इंसुलिन मेजर करोगे तो वो अंदर बॉडी में जो प्रोड्यूस होता है वो इंसुलिन बताएगा या जो बाहर से देंगे हम वो इंसुलिन बताएगा. सो टू डिफरेंशिएट बिटवीन एंडोजीनियस एंड एक्सोजीनियसली एडमिनिस्टर्ड इंसुलिन, सी पेप्टाइड लेवल्स आर मेजर्ड. सी पेप्टाइड बताता है कि अंदर बॉडी में जो प्रोड्यूस हुआ है इंसुलिन उसका मात्रा कितना है. C peptide. So this is connecting peptide A chain and B chain. 21 amino acid in the A chain, 30 amino acid in the B chain. So in the A chain, there is 21 amino acid, B chain there is 30 amino acids, and it is stabilized by interchain and intrachain disulfide linkages. Interchain and intrachain disulfide linkages. Insulin is a polypeptide. It is a branch polypeptide. It is stabilized by the disulfide linkages. Initially it is produced like this. So this is polypeptide, primary structure of protein, branch protein. So insulin was isolated by Banting and Best, and they got Nobel Prize. Banting and MacLeod got the Nobel Prize. Sanger got the Nobel Prize for the discovery of the structure, means amino acid sequence of the insulin. So there are two chains, A and B chain. A has 21 amino acid, B has 30 amino acids. Disulfide linkage occur by disulfide group of cysteine. Okay, so in between the 7th of A and B chain there is disulfide linkage. In between 20 and 19th, 20 of the A chain and the 19th of the B chain, there is disulfide linkage, and in between the 6th and 11th of the A chain, there is intrachain intrachain disulfide linkage. These are the interchain disulfide linkages. So this is the structure of insulin, primary structure. Remember this. This is the structure of insulin. Now important short note, SAQ. SAQ, alpha helix. Important short note, alpha helix. So secondary structure. Further coiling of primary structure upon itself leads to the formation of secondary structure. There are 5 examples of secondary structure: alpha helix, beta pleated sheet, turn, bends and loops. क्या क्या बोला मैंने? Alpha helix, beta pleated sheet, turn, bends and loops. So these are the 5 example of secondary structures of protein, of which alpha helix is the most common and it is important. Alpha helix is the most common and it is important. Alpha helix, it is most common. It is stable conformation. It is right-handed. The most common amino acid in the alpha helix is methionine, followed by glutamate. Most common is the methionine. The least common amino acid is the proline. Least common amino acid is the proline in the alpha helix structure. Now alpha helix, see, alpha helix, it is right-handed, stable, spiral. It is stable, spiral, right-handed. Most common amino acid is methionine, followed by the glutamate. Least common is the proline. The coiling, why the name alpha is given? Because Pauling and Corey ने जो भी स्ट्रक्चरल क्लासिफिकेशन दिया उसमें ये सबसे पहले मिला है उनको, इसलिए उसको अल्फा दिया, जो बाद में मिला उसको बीटा दिया, अल्फा बीटा. Okay, so most common amino acid is methionine, least common is the proline. Now this is a turn of alpha helix, which is which contain 3.6 amino acids per turn. Each turn of alpha helix contain 3.6 amino acid. 3.6 amino acid. Pitch of the turn is 0.54 nanometer. 0.54 nanometer, and distance between two amino acid is 0.15 nanometer. Distance between two amino acid is 0.15 nanometer, and it is stabilized by. So this is stabilized by mainly, the most important in the stabilization is the hydrogen bond. The intra-chain hydrogen bond is important here. So intra-chain hydrogen bond is responsible for stabilization of secondary structure. Now see, it is between the first and fourth amino acid. It is between the carbonyl, oxygen and amide nitrogen. Carbonyl, oxygen and amide nitrogen, there is hydrogen bond, and it is between the first and fourth. See this is hydrogen bond. So this is hydrogen bond. See this, it is first and fourth amino acid. So if it is first, it is fourth amino acid. Carbonyl, oxygen and amide hydrogen, that is, it forms the hydrogen bond, and intrachain hydrogen bonds, they are important in the stabilization of alpha helix. So least common amino acid is proline, and the amino acid which disrupt the alpha helix, it includes the proline, it includes the glycine, it includes the series of acidic amino acid and series of basic amino acid that disrupt the alpha helix. Example of alpha helix is there in the hemoglobin molecule. There is alpha helix. Hemoglobin is a quaternary structure, but each chain of the hemoglobin, it has the primary, secondary and tertiary structure. So it is alpha helix, it is important in the hemoglobin and myoglobin structure. So example, it is hemoglobin, alpha helix in the hemoglobin and myoglobin. There are 8 alpha helices in the myoglobin structure. Okay, so now this is all about the alpha helix. So primary structure हमने discuss किया, secondary में alpha helix हमने discuss किया. Right-handed, spiral, most common, stable conformation. Each turn contain 3.6 amino acid. The pitch of each turn is 0.54 nanometer. Distance between two amino acid is 0.15 nanometer. There is a hydrogen bond, intrachain hydrogen bond, which is important for the stabilization of secondary structure. It is between the carbonyl oxygen and the amide nitrogen. Carbonyl oxygen and amide nitrogen, there is hydrogen bond which is responsible for stabilization of alpha helix. Example, alpha helix, it is present in the structure of hemoglobin and it is present in the structure of myoglobin. Hemoglobin is the quaternary structure, but and myoglobin is the tertiary structure, but this secondary alpha helix structure is there in this quaternary and tertiary structure. So this is all about the alpha helix. Now beta pleated sheets, there are two types of beta pleated sheet. These are the extended in zigzag manner. Beta pleated sheet, extended polypeptide in zigzag manner. There are two types, parallel and anti-parallel beta pleated sheet. Parallel and anti-parallel beta pleated sheet. Now see, this is C terminus, this is N terminus, this is N terminus, this is C terminus. Okay, so it is C, C, N, N. So parallel beta pleated sheet. And if it is C terminal here and N terminal below this, it is the anti-parallel beta pleated sheet. Parallel and anti-parallel beta pleated sheet. Pauling and Coren ने जब discuss किया, this was second in the discovery, that's why name was given, that is beta pleated sheet. There are two types of beta pleated sheet, parallel beta pleated sheet and anti-parallel beta pleated sheet. Most common amino acid in the beta pleated sheet is the valine. Most common amino acid is the valine, and beta pleated sheet, they are stabilized by, this is, see, this is interchain, this is interchain hydrogen bonds, 6 carbonyl oxygen and amide nitrogen. There is formation of hydrogen bond in between the two chains. So interchain hydrogen bonds, they stabilizes the beta pleated sheets. Example is the carbonic anhydrase enzyme. Example is carbonic anhydrase enzyme. It contains both parallel and anti-parallel beta pleated sheets. So parallel beta pleated sheet, anti-parallel beta pleated sheet. Now third one is, just wait, third one is the bends, turns and loops. So turns, bends, this is short polypeptide, which links to secondary structure, to helices, which links to helices. So short peptide links to helices, turn and bends, and the loops, these are the long peptide which connect to secondary structure, which connects secondary structures, they are known as loops. So turn, bends and loops, they are connecting peptide. They are important for the connection of two secondary structures. Now see this. So this is secondary structure, alpha helix, beta pleated sheet, turn, bends and loops. Now the tertiary structure. Now see, tertiary structure, example is myoglobin. Myoglobin is example. Myoglobin, it has 8 alpha helices and iron at its center. Myoglobin, what is the role of myoglobin? It is important in the storage of oxygen. Myoglobin, important in the storage of oxygen. Myoglobin, 8 alpha helices with iron at its center. 8 alpha helices with iron at its center. What do you mean by tertiary structure? The folding of the secondary structure and formation of or assembly into a three-dimensional protein, functional protein, is known as the tertiary structure of the protein. What are the bonds which are important in the tertiary structure of protein? It is the hydrogen bond between the R group. It is the hydrogen bond between the R group. It is ionic interaction, that is positive and negatively charged group. It is the hydrophobic interaction and the van der Waals forces. Van der Waals forces, these are the weak forces which stabilizes the tertiary structure of the protein. So here example is myoglobin. What are the bonds responsible for stabilization of tertiary structure? It is the hydrogen bond between the R group. Second, hydrophobic interaction between the hydrophobic amino acids. Hydrophobic means non-polar amino acids, that is valine, isoleucine, leucine, or phenylalanine, tyrosine, tryptophan. These are the non-polar amino acids. Then the ionic bond between the positively and negatively charged amino acids. It will stabilizes the tertiary structure. Also the van der Waals forces. So, एक, दो, तीन, ionic bond तीन, and the fourth one is the van der Waals forces. Van der Waals forces that stabilizes the tertiary structure of protein. Example is myoglobin. Example is myoglobin. So tertiary structure of the protein. This diagram is important. So here the hydrophobic interaction between the non-polar amino acids, the positively and negatively charged group, they forms the ionic bond. The hydrogen bond between the R groups and the van der Waals forces, they stabilizes the tertiary structure of protein. Example is myoglobin. हमने बोला था self constructed room is a example of tertiary structure of the protein. Myoglobin is the example. And this is quaternary structure. Quaternary structure. When polypeptide is 2 or more than 2, number polypeptide number, it is 2 or more than 2. It if it is 2 or more than 2, it is quaternary structure of protein. Hemoglobin. So oxygen molecule, it carries the oxygen molecule. It has 2 alpha chain, 2 beta chains in the hemoglobin. 2 alpha chain, 2 beta chain. Hemoglobin molecule, quaternary structure of protein. 4 units are there, 2 alpha chain, 2 beta chain. It is important in carrying the oxygen to the tissues. Quaternary structure of protein. So this is all about the structural organization of protein. Important LAQ long answer question in the university examination from chemistry of proteins. From the chemistry of protein. Primary, secondary, tertiary, quaternary structure. Number and sequence of amino acid is important in the primary structure. Folding of primary structure upon itself form the secondary structure. Then the assembly of the amino acids into three-dimensional structure form the tertiary structure, and when it is units, more than 2 units are there, it is known as the quaternary structure. So single polypeptide chain can be oriented into primary, secondary, and tertiary structure. If there is 2 or more than 2 polypeptide chain, it indicates the quaternary structure of protein. So this is all about the structural organization of protein. हाय शेखर, स्पैम मत करो. ओके. हेलो ऑल. एग्जाम में स्ट्रक्चर बनाना इंपॉर्टेंट है. एग्जाम में फ्लो चार्ट्स डायग्राम इंपॉर्टेंट है. फ्लो चार्ट्स डायग्राम इंपॉर्टेंट है. सी ये जो क्वाटरनरी स्ट्रक्चर ऐसा भी नहीं बना पाए आप, ऐसा भी बनाए तो भी बनाओ, लेकिन बनाना तो पड़ेगा कुछ ना कुछ. तो ऐसा भी बनाया तो भी चलेगा. क्वाटरनरी स्ट्रक्चर, टर्शरी स्ट्रक्चर एकदम इजी है. मैं बताता आपको. टर्शरी स्ट्रक्चर, इट विल गिव द इम्पैक्ट. टर्शरी स्ट्रक्चर आप निकालोगे, इट विल गिव द इम्पैक्ट. सो ड्रा लाइक दिस. This is tertiary structure. It will give you the impact. Now see here, the CH3 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 CH3. So this is hydrophobic interactions. Hydrophobic interaction. Okay. So this is the bonds which are responsible for stabilization of tertiary structure. Then see CH2 NH3 positive, and O, it is negative CH2. So this is ionic bonds. Ionic bond, which is important here. Then the See, this is hydrogen bond. This is disulfide linkage. Disulfide linkage. So in that way, draw the diagram. So flowchart diagrams, they are important while writing your answers. So this is tertiary structure of protein. Tertiary structure of protein. See, this hydrogen bond, hydrophobic interaction, ionic bond, disulfide linkage, and van der Waals forces. They are important in stabilization of tertiary structure. They are important in the stabilization of quaternary structure. And intrachain hydrogen bonds, they are important in the stabilization of secondary structure of the protein. Peptide and occasional disulfide bond, they are important in the primary structure of proteins. Okay. Now we'll see the plasma proteins. Important short note, plasma proteins. So plasma protein C, this is ये ऐसी हमको डायग्राम निकालनी है, 1 2 3 4 5 ऐसी डायग्राम निकालनी है. सी, सी ऐसी मुट्ठी हमको बंद करके ऐसे डायग्राम निकालनी है. दिस इज अल्बुमिन, दिस इज अल्फा 1, दिस इज अल्फा 2, बीटा एंड गैमा ग्लोबुलिन. Plasma proteins, it is important short answer question, and it is also connecting with the three important clinical cases: cirrhosis of liver, multiple myeloma, and nephrotic syndrome. Cirrhosis of liver, multiple myeloma, and the nephrotic syndrome. Three important cases, electrophoretically important. Okay. Plasma proteins, now see, this is plasma proteins. Just wait, एक फिर से निकालते हैं हम. So now see, this is albumin band. Okay, this is albumin. This is albumin band. This is alpha 1 globulin. This is alpha 2. This is beta. This is gamma globulin. Albumin, alpha 1, alpha 2, beta, and gamma globulin. So we are in the plasma protein as an important short note. Now, what is albumin concentration? The concentration of albumin is 3.5 to 5.0 gram per DL. तो ये अकेला पैरामीटर है जो बायोकेमिस्ट्री के प्रैक्टिकल करते वक्त जो ग्राम पर डीएल में है, प्रोटीन एंड अल्बुमिन, टोटल प्रोटीन एंड अल्बुमिन. ओके, बाकी सब मिलीग्राम में है, और जो भी बचे हुए एंजाइम्स है दैट इज इन द इंटरनेशनल यूनिट पर लीटर. सो हाउ टू रिमेंबर द यूनिट? ओनली अल्बुमिन, टोटल प्रोटीन, इट इज इन द ग्राम. Rest of the parameters, it is in the milligram per deciliter, and the enzymes, they are in the international unit per liter. So albumin concentration, it is 3.5 to 5.0, or some book 5.5 it is also given, gram per deciliter. Now alpha 1, alpha 2, beta, gamma, it is not necessary to remember, but it is easy. See, it is 0.3 to 0.6 gram per dl. It is 0.4 to 0.8 gram per dl. It is 0.6 to 1.1 gram per dl, beta globulin, and this is zero point 0.8 to 1.8 milligram per deciliter. This is in milligram. Globulins, it is in the milligram. These are the plasma proteins. These are the plasma proteins. Okay. Albumin, alpha 1, beta, alpha 1, alpha 2, beta, and gamma globulins. So albumin concentration, it is 3.5 to 5.0 gram per deciliter. Globulins, it is 2.5 to 3.5 gram per deciliter. And fibrinogen, it is 200 to 300 milligram per deciliter. It is in milligram. This is in gram per dl. This is in gram per dl. Okay, so these are the plasma proteins and their levels. It is very very important for diagnosis of various diseases, particularly the electrophoretic pattern is important. When serum is utilized for the electrophoresis, you will get these five bands: albumin, alpha 1, alpha 2, beta and gamma. But when you use the fibrinogen, when we use the fibrinogen, you will miss when when we use the plasma. In the plasma there is fibrinogen, so you will get band of this additional band of this fibrinogen in the plasma, and this can be confused with the band of multiple myeloma. तो हमने कौन सा सैंपल लगाया, सीरम लगाया क्या प्लाज्मा लगाया, वो इंपॉर्टेंट है. रिमेंबर, प्लाज्मा प्रेजेंट फाइब्रिनोजेन. व्हेन वी यूज सीरम, सीरम में क्या होता है, क्लॉटिंग के बाद सीरम ऊजेस आउट. नाउ द वो जो फाइब्रिनोजेन है सीरम में, वो जो क्लॉटिंग के टाइम में वो यूटिलाइज हो गया. It is utilized during the clotting process, fibrinogen. So remember, plasma present fibrinogen. Plasma में fibrinogen present रहता, सीरम में वो present नहीं रहता, वो clot formation में utilize हो जाता, इसलिए वो सीरम में नहीं रहता. So you will get these type of bands. Now there are albumin. What are the functions of albumin? All of you know the functions of albumin. So it is important for colloidal osmotic pressure. Colloidal osmotic pressure. It is important for colloidal osmotic pressure. It is important for the nutrition. It is important for the buffer mechanism. Buffering action, it is important. And it is important, albumin, it is important for the transport of drugs, transport of steroids, transport of other hormones, transport of ions, transport of free fatty acid. It is important. So albumin has important function in our body. It is important for the colloidal osmotic pressure. Decrease albumin level causes the edema, swelling, edema, decrease albumin level. So it is important for the colloidal osmotic pressure. It is important for the nutrition. It is having the buffering action. It is important for transport of the free fatty acid, steroids, bilirubin, drugs, ions. It is very very important. This is very important function of albumin. Alpha 1, see now next slide. So alpha 1 globulins examples, it includes alpha 1 antitrypsin. This accounts 90% of total alpha globulins. Then HDL, alpha lipoprotein HDL. Alpha 1 antitrypsin, it is important. It is also known as alpha 1 antiproteinase, and it inhibits the various antiproteinase. It inhibits the serine proteases. It inhibits the trypsin. So this is function alpha 1 antitrypsin, which accounts for 90% of alpha globulin. HDL, high density lipoprotein. It is important for reverse transport of cholesterol from peripheral tissues to the liver, and it is important for metabolism of cholesterol. So it is cardioprotective. So that's why it is also known as good cholesterol. HDL, alpha 1 antitrypsin, HDL. Then the thyroid binding globulin, thyroid binding globulins, retinol binding globulins, retinol binding globulins. Then orosomucoid. These are the examples of alpha 1 globulins. These are the examples of alpha 1 globulins. Alpha 1 globulins. The concentration is 0.3 to 0.6 gram per dl. 0.3 to 0.6 gram per deciliter. Alpha 1 globulins. Now alpha 2. Alpha 2. So alpha 2 globulins, it includes alpha 2 fraction, alpha 2 globulins. It includes alpha 2 macroglobulin. It is larger in size, and that's why it is not filtered in patient with glomerulonephritis. It is retention of occur, and there is increase in alpha 2 fraction in the nephrotic syndrome. So alpha 2 macroglobulin, then haptoglobin. Haptoglobin, it binds with the extracorpuscular hemoglobin and prevent the excretion of hemoglobin molecules. So haptoglobin, prothrombin, important in the clotting process, prothrombin, and ceruloplasmin. Ceruloplasmin has two important role. First, it is important. Ceruloplasmin, it converts ferrous into ferric state, and it incorporates ferrous into the transferrin molecule. That is, for that function ceruloplasmin is important. Ceruloplasmin, it is important. Okay, ceruloplasmin, it is decreased level of ceruloplasmin. It occurs in the, it is important for the copper transport, and it is important for conversion of ferrous into ferric state, and its incorporation into the apotransferrin, so that it is converted into ferrous is converted into ferric, and it is incorporated into transferrin molecule. The levels of ceruloplasmin decreases in the Wilson's disease, which is related with the copper metabolism. So these are the alpha 2 globulins. Alpha 2 globulins. Here the alpha 2, this is macroglobulin. It is important. It is also having the plasminogen, inhibits the plasminogen. It inhibits the trypsin. It is having the antitrypsin like activity, alpha 2 macroglobulin. It is larger in size, and retention occur in the nephrotic syndrome, alpha 2 globulin. So alpha 1, alpha 2, then beta, then the beta. So beta globulins, it includes LDL, low density lipoprotein, which is also known as bad cholesterol. The increased level of LDL and their oxidation with the help of free radical, it is responsible for the atherosclerosis. So low density lipoprotein, it is beta lipoprotein, beta globulin, LDL, then hemopexin, which is important for the transport of the heme, then plasminogen, and transferrin. These are the beta globulins. And last one is the gamma, that is gamma, and it includes the gamma globulins. That is antibodies. So it includes IgG, AMDE. These are the antibodies, immunoglobulins. So this is how they are classified, plasma proteins. So plasma protein, important short note, albumin globulins, alpha 1, alpha 2, beta, gamma globulins, uh, alpha 1, alpha 2, beta, and gamma globulins. These gamma globulins, they are produced by the B cells. They are produced by the plasma cells. They are gamma globulins. They are produced by the plasma cell in response to antigen. They are also known as antibodies. Immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and E. Depending upon the heavy chain type, they are classified into G, A, M, D, E. Immunoglobulins, various types of immunoglobulin. So these are the antibodies, gamma globulin. So this is how plasma proteins are classified. Now three classical disorders, they are important here. ये है क्या? Just wait. So disorders, they are important here. See, in the, this is normal. This is normal pattern of electrophoresis. This is positive terminal. This is negative terminal. In the electrophoresis, the protein in the pH 8.6, means alkaline pH, protein acquires negative charge and move towards the positively charged electrode. What do you mean by electrophoresis? It is movement of charged particle under the influence of electric field towards the oppositely charged electrode. So this is how the normal pattern looks like. Now, in the cirrhosis of liver, liver, cirrhosis, of liver, there is decrease in the albumin level occurs, alpha 1, alpha 2, beta, and there is increase in the gamma, or sometime, just wait. So what happens in the cirrhosis of liver, there is decrease. This is normal. This is normal electrophoretic pattern. Now in the cirrhosis, there is decrease albumin band because synthesis of albumin occurs in the liver. So there is decrease albumin band, and beta gamma bridging may be there. Beta gamma bridge will be there. So there is increase in the compensatory gamma globulins. Compensatory increase in the gamma globulins in the cirrhosis of liver. So this is pattern. See, so there is decrease in the albumin level, alpha 1, alpha 2, beta, alpha 1, alpha 2, beta, and gamma. So beta gamma bridge may be there in the cirrhosis of liver. There is increase in the gamma globulins, and there is decrease in the albumin level in the cirrhosis of liver in the electrophoresis pattern. This is one pattern, that is cirrhosis of liver. Then second, nephrotic syndrome. In the nephrotic syndrome, there is decrease in the albumin occurs, alpha 1, there is increase, alpha 2, slight increase in the beta, and normal gamma globulin. So there is decrease albumin level because albumin excreted in the urine in the patient with nephrotic syndrome because of loss of negatively charged heparan sulfate in the glomerular basement membrane. So selective permeability जो रहती वो loss हो जाती, albumin excrete होता यूरिन में तो albumin level decrease हो जाती, and there is retention of alpha 2 occurs. Alpha 2 increases, retention occur. This is because of its larger size, retention of alpha 2, and to some extent beta also retention occurs. So there is prominent alpha 2 and beta band, and this is gamma. Okay, so there is in the nephrotic syndrome, there is decrease albumin level, prominent alpha 2 globulin because of large size of alpha 2 macroglobulin. This may be asked in the viva because of larger size of alpha 2 macroglobulin. This is also asked in the uh, quiz question, why alpha 2 retention occurs in the nephrotic syndrome. So it is because of larger size of alpha 2 macroglobulin. Okay, so this is nephrotic syndrome. And in the third is the multiple myeloma. So in this multiple myeloma, there is decrease in the so this is prominent M band in the electrophoresis. There is decrease albumin level. This is multiple myeloma, plasma cell tumor. Multiple myeloma, plasma cell tumor characterized by punched out lesion on the bone. There is characteristic M band because of monoclonal gammopathy. Only single types of immunoglobulins they are produced. Only single. This is monoclonal gammopathy. Single type of cellular responses there. Single type of immunoglobulins they are produced, and that gives sharp band in the electrophoresis. So M band is there, and there is decreased albumin level in the multiple myeloma. Okay, so इतना डिक्रीज नहीं होगा, जस्ट वेट. So it is the prominent M band in electrophoresis. It is a feature of multiple myeloma. Single types of immunoglobulins they are produced. Single type of immunoglobulins they are produced. That's why there is sharp M band. It is not diffuse gamma band. It is sharp M band in the electrophoresis in the multiple myeloma. So you may get characteristic electrophoretic pattern in your practical examination, in the laboratory technique interpretation. These three: nephrotic syndrome, multiple myeloma, and the cirrhosis of liver. These are classical features in the electrophoresis. So this is important topic for the practical examination also, viva question also, electrophoretic technique and their interpretation, it is important. So it is important. So nephrotic syndrome, multiple myeloma. Okay. Now we'll discuss the important question, next important question, that is biologically important peptides. Hi Snehajit. Hello all of you. So next question, next important short note is biologically important peptide. Now see, peptides means less than 10 amino acids, less than 10 amino acid count, it is less than 10, it is known as the peptide, biologically important peptides, and their function, it is important here. So biologically important peptides, it includes suppose tripeptide is there, tripeptide means 3 amino acids, 2 peptide linkages, 3 amino acid, 2 peptide linkages. Dipeptide, dipeptide means 2 amino acids, like that. Dipeptide, tripeptide, tetrapeptide, pentapeptide, heptapeptide, octapeptide, nanopeptide, and decapeptide. When there is less than 10 amino acids are there, it is known as peptide. And biologically important peptide, it includes some important peptides we'll discuss. For example, C, glutathione. Glutathione, it is tripeptide. Glutamyl, cystinyl, glutamyl, cystinyl, glycine, gamma glutamyl, cystinyl glycine, important. Glutathione, important tripeptide. It is tripeptide, tripeptide. Then again TRH, thyrotropin releasing hormone. It is tripeptide. So glutathione and thyrotropin releasing hormone, they are tripeptide. Glutathione, it is important for the buffer, it is important for maintaining the RBC integrity, it is important in the that is free radical scavenging action. Glutathione has the important role. Reduced glutathione, it is important for the maintaining the RBC integrity. So this is very very important. Glutathione, glutamyl cystinyl glycine. Also it is important in the Mr cycle, which is important for transport of amino acid, transport of amino acid in the renal tissues, in the brain renal tissues, it is important, glutathione. So this is about the glutathione. Thyrotropin releasing hormone, it is important for the release of the thyroid hormones, important for the release of thyroid hormone. Then the ADH, antidiuretic hormone, vasopressin, it is important. Vasopressin, it is important. It is nanopeptide, contain 9 amino acid, nanopeptide. And again one more, that is oxytocin, it is important. It is also nanopeptide, 9 amino acids, nanopeptide. Oxytocin, vasopressin, it is important for the water retention, maintenance of the blood pressure. Oxytocin, it is important for the uterine contraction. It is also used as a drug for the parturition, for the uterine contraction. It is also important for the milk, secretion of the milk. Oxytocin is important. So uterine contraction, it is important. Oxytocin is important. So vasopressin, oxytocin, these are the nanopeptide. Then angiotensin 1, angiotensin 1, which is converted into angiotensin 2, angiotensin 2. So renin angiotensin system, which is important for the release of aldosterone and the maintenance of the blood pressure. It is important. Angiotensin 1, angiotensin 2, it is important. So it is decapeptide, 10 amino acids are there, decapeptide, and this is octapeptide. octapeptide. So this is important, octapeptide and decapeptide. So this conversion, one is converted into two by the angiotensin converting enzyme ACE, and there are group of drugs which act on this angiotensin converting enzyme, and they are the first choice of drug for the treatment of hypertension, captopril, enalapril. These ACE inhibitors, they are used in the treatment. So they are antihypertensive drug, captopril, enalapril, ACE inhibitors. They are used to inhibit this. So this is decapeptide, this is octapeptide. Then calidin, it is converted into bradykinin. Bradykinin, these are the vasodilator. This is vasodilator, vasodilator. So it is important. These are also peptides. Then some hormones, gastrin, secretin. Then some antibiotic, synthetic antibiotic, that is gramicidin, bacitracin. These are the biologically important peptide. Dipeptide, दो amino acid, carnosine, anserine, aspartame. Aspartame is the artificial sweetener. Carnosine, anserine, aspartame, dipeptide. Tripeptide, glutathione, thyrotropin releasing hormone. Pentapeptide, enkephalin, act like opiate. They are natural analgesic. Pentapeptide, क्या बोला मैंने, enkephalins. Then the octapeptide, that is angiotensin 2. Nanopeptide, vasopressin, oxytocin, and their uses. Then decapeptide, that is calidin. Then this bradykinin is also important. It causes the vasodilation. So these are the important, biologically important peptide and their functions. So this is also a important short note, biologically important peptides. Now the immunoglobulins. Immunoglobulins, this is important question again. Immunoglobulin, their structure and their function. This topic is particularly important for the MCQs. It is important for the BAQ, short answer question. It is also important. Immunoglobulin, their structure, their type, and their characteristics. So we'll discuss this in short. Immunoglobulin, see. So immunoglobulin structure, they are composed of the heavy chains, two heavy chains and two light chains are there. Two heavy chain, it is Y-shaped structure, it is Y-shaped tetramer, it is Y-shaped tetramer, two heavy chains, and depending upon the heavy chain types, they are classified into, for example, they are classified as IgG, A, M, D and E. IgG, A, M, D, E. So immunoglobulin G, immunoglobulin A, M, D, E. Depending upon the class of heavy chain, it is the gamma in the IgG, it is alpha in the A, it is mu in the M, it is delta, and it is epsilon. So these are the types of heavy chain in this type of immunoglobulin. So depending upon the type of heavy chain, they are classified as G, A, M, D, E. Now the light chain, it is mainly kappa or lambda. Kappa or lambda, most common it is kappa 60%, this is 40%. 60% kappa or lambda, never both. So it is composed of heavy chain and light chain, heavy chain and light chain. Each heavy chain has 3 constant region and 1 variable region. So this is variable region of the heavy chain, this is variable region of the light chain, this is constant region of the light chain, this is constant region of the heavy chain, constant region of heavy chain, constant region of the heavy chain. Okay, so these are the different types of immunoglobulins. Y-shaped structure, tetramer, it is tetramer, and it is these antibodies, they are produced by the plasma cell in response to various antigen. So special characteristic, IgG, immunoglobulin G, there are 4 types, G1, G2, G3, G4, 1, 2, 3, 4. So there is variation in the amino acid sequence. So it is IgG 1, 2, 3, 4. Now only except G2, all this IgG crosses the placenta. IgG crosses the placenta. Immunoglobulin G, it is important for the secondary immune response. IgM, immunoglobulin M, it is for the primary immune response. This is for the primary immune response. immune response. This is secondary immune response. Immune response. Then this M, it can be a monomer, it can be a pentamer, pentameric molecule. 9 lakh is the molecular weight of M. It is millionaire molecule 9 lakh is the molecular weight. It can be a pentameric, and these units, these pentamers, they are joined by the J chain. So there is presence of J chain in the M IgM molecule. Okay, so this is millionaire molecule. This crosses the placenta. This is important for the opsonization, means phagocytosis. This is important for the complement fixation by the classical pathway. IgG important for the complement fixation. IgM is also for the complement fixation, and it is also responsible for the opsonization, means phagocytosis. Then IgM, it is millionaire molecule. It is act on the B cell surface, act as a B cell surface receptor. Also, immunoglobulin D act as a BC cell, means cell surface receptors on the B cell. Okay, so see, immunoglobulin G crosses the placenta. So immunoglobulin G crosses placenta. Just wait. Placenta except G2. It is important in opsonization. It is important for the complement fixation. It is having highest serum concentration. Then IgG, it is important for the secondary immune response. Secondary immune response. IgG. Now IgM. See, IgA. Take A. There are two types. That is secretory IgA, which is present in the various secretions. It is present in the colostrum, it is present in the milk, it is present in the tears, saliva. Secretory IgA. And second one is the serum IgA. Serum IgA, it is monomeric unit, while this is dimeric unit. And it gives the mucosal immunity. Prevent from the various infections. IgA. Immunoglobulin A, the fix the complement by the alternate complement pathway. So this is immunoglobulin G, this is immunoglobulin A. Then the immunoglobulin M. It is millionaire molecule. The molecular weight is much more as compared to other immunoglobulins. Pentameric. Monomeric are pentameric and connected by the J chains. Also there is J chains in the immunoglobulin A. Secretory IgA, there is J chain. Then it is opsonization. It is important for opsonization, more important than the immunoglobulin G. It is important for the complement fixation, more than the immunoglobulin G, because it is pentameric. So it is having the valency 10. It can combine with the 10 antigen molecules. So it is having the valency 10. Pentameric, J chain, opsonization is there. It is responsible for the complement fixation, and it is responsible for primary immune response. And it is present on the B cell, act as a B cell receptor. Act as an B cell receptor. So IgM. Then IgD, immunoglobulin D. It is having the highest carbohydrate content, and it acts as a B cell receptor. And the most important for MCQ is immunoglobulin E. It is immunoglobulin E. It is responsible for allergic responses. It is important in worm infection. It is responsible for the hypersensitivity reaction. Allergic responses, worm infection. It is least concentration in the blood, least concentration, present in the least concentration. So these are the some properties of the immunoglobulin. So this can be a, this is important in the viva, this is important in the MCQ part of the biochemistry. So immunoglobulins. Now we'll start the protein metabolism. This is again very important. Digestion and absorption of protein and the protein metabolism, that includes the urea cycle, ammonia metabolism and urea cycle, that includes the phenylalanine and tyrosine metabolism, that includes the tryptophan metabolism, that includes the sulfur containing metabolism and metabolism of glycine. So we'll discuss the important topics from the protein metabolism. Just wait 2 minutes. ले लो भाई, 5 मिनट का ब्रेक ले लो. So these antibodies, they present themselves on the surface of various cell, and they are responsible for the opsonization, phagocytosis. Hi Irfan. ऑप्सोनाइजेशन मतलब एंटीबॉडीज का कोटिंग कर देते वो ऑर्गेनिज्म के सराउंडिंग में, और वो फिर फैगोसाइटोज हो जाता, उसको उसको पकड़ लेते फैगोसाइट्स, उसको बोलते हैं ऑप्सोनाइजेशन. मत देखो भाई, नीट यूजी स्टूडेंट्स क्यों देख रहे हैं. सी, अब तक हमने क्या क्या कवर किया. प्रोटीन एंड अमाइनो एसिड केमिस्ट्री के इंपॉर्टेंट क्वेश्चंस कवर किए हमने. 3 इंपॉर्टेंट क्लिनिकल केसेस कवर किए, दैट इज नेफ्रोटिक सिंड्रोम, मल्टीपल माइलोमा एंड द सिरोसिस ऑफ लीवर. फिर हमने इसके शॉर्ट नोट्स कवर किए. हमने कौन से शॉर्ट नोट कवर किए? हमने प्लाज्मा प्रोटीन्स कवर किया, फंक्शनल क्लासिफिकेशन ऑफ प्रोटीन कवर किया, अमाइनो एसिड क्लासिफिकेशन कवर किया, फिर बायोलॉजिकली इंपॉर्टेंट पेप्टाइड कवर किया, फिर इम्यूनोग्लोबुलिन्स कवर किए. फिर हमने एक LAQ किया, कवर किया, दैट इज स्ट्रक्चरल ऑर्गेनाइजेशन ऑफ प्रोटीन, दैट इज प्राइमरी, सेकेंडरी, टर्शियरी एंड क्वाटरनरी स्ट्रक्चर ऑफ प्रोटीन. ये इतने सब क्वेश्चंस हमने, जो इंपॉर्टेंट टॉपिक्स है, इंपॉर्टेंट क्या, लगभग सारे ही टॉपिक हमने कवर किए जो प्रोटीन केमिस्ट्री में इंपॉर्टेंट है, अमाइनो एसिड एंड प्रोटीन केमिस्ट्री में इंपॉर्टेंट है. Now we will start with the metabolism. There are number of cases in the protein metabolism, starting with the phenylketonuria, alkaptonuria, homocystinuria, maple syrup urine disease, pheochromocytoma, Parkinson's disease, कार्सिनाइड सिंड्रोम, प्राइमरी हाइपरऑक्सालूरिया. बहुत सारे इंपॉर्टेंट केसेस है, और वो इंपॉर्टेंट शॉर्ट नोट्स भी है. ऑल्सो वो इनबॉर्न एरर्स भी है, वो पर्टिकुलर अमाइनो एसिड से रिलेटेड, तो वो इंपॉर्टेंट है. So the first and the priority topic in amino acid metabolism is the ammonia metabolism and urea cycle with disorders. ये बहुत important है, first priority topic है, first priority topic है. Okay, now we'll start with the digestion and absorption of protein. Here, see, we'll draw one flow chart in the digestion and absorption of protein. So see, two types of enzymes, they work on the protein, the endopeptidases and exopeptidases. Endo मतलब जो अंदर के internal peptide bonds को क्लीव करेंगे, exo मतलब जो बाहर के peptide bonds को क्लीव करेंगे. Endopeptidases and exopeptidases, they are used in the digestion and absorption of the protein. Now when we take the protein in the mouth, digestion is nil in the mouth. Digestion of protein, it is nil in the mouth. So no digestion in the mouth. When the protein enters the stomach, when it enters the stomach, there is, in the stomach there are three things which are important in the digestion of protein. The first one is, the first in the digestion of protein is HCl, hydrochloric acid. Second one is the pepsinogen, which is converted into pepsin, and third one is the renin, which is important in the infant. Renin, it is important in the infant. So HCl, it causes the denaturation of protein, denaturation of the protein. HCl causes the denaturation of protein, and this denaturation, it exposes this denaturation and expose the peptide bond for action of the pepsin, for the action of the pepsin. This is important function of the HCl. Second important function of the HCl is it converts this pepsinogen, activates the pepsinogen into pepsin, This is important function of HCl in the stomach. So stomach में तीन चीजें हैं, HCl, pepsin and renin. So pepsin will act on the N-terminal peptide bond. N-terminal, not N-terminal, just wait. It act on the peptide bond, it cleave the peptide bond formed by the mainly aromatic amino acid, that is N-terminal side, phenylalanine, tyrosine, tryptophan, leucine and methionine. N-terminal peptide, it act on the peptide bond which is formed by the N-terminal phenylalanine, tyrosine, tryptophan, leucine and methionine. Peptide bond, pepsin, it is active at the acidic pH 1 to 2 pH. And renin, it converts the casein, milk casein, it is milk casein, just wait, milk casein, which is converted into calcium paracasinate curd, insoluble curd, so it increases the transit time of the food, transit time of milk in the stomach, increases the transit time so that it is available for the action of the various enzymes. So this is action of the renin. So this is in the stomach, this occurs in the stomach. Mouth, there is nil, digestion nil. Stomach, there is action of these three compound, HCl, pepsinogen is converted into pepsin. Pepsin also itself catalyzes the conversion of pepsinogen into pepsin. It acts on the peptide bond, it is endopeptidase, cleave the internal peptide bond and converts the larger polypeptide molecule into the smaller one. So and the renin, which is present in the infant, which converts the milk casein into calcium paracasinate insoluble curd and increase the transit time. So mouth, stomach, then intestine, there is release of pancreatic enzyme. When food bolus reaches the intestine, when it reaches the intestine, there is release of cholecystokinin, there is release of secretin, and secretin. So cholecystokinin causes release of pancreatic enzyme and secretin causes the release of bicarbonate, which neutralizes the pH of this acidic pH, which neutralizes. So pancreatic enzyme, there is secretion of pancreatic enzymes here. So there is pancreatic enzymes. So which are the pancreatic enzymes? Are endopeptidase mainly. So pancreatic enzymes, it includes trypsin, enterokinase, which converts the trypsinogen into trypsin. Then elastase, just wait, trypsin, after trypsin, chymotrypsin. It is chymotrypsin elastase. So trypsin activates the chymotrypsin and elastase and the carboxypeptidase. Carboxypeptidase. These are the pancreatic enzyme peptidase. So these are the pancreatic enzyme. This trypsin will act on the arginine and lysine, the peptide bond which is formed by the arginine and lysine. These are the endopeptidase. This is exopeptidase which act on the carboxyl terminal amino acid. Carboxyl terminal पे जो amino acid है उस पे act करेगा carboxypeptidase. Okay, trypsin, chymotrypsin, elastase. This will act on the peptide bond formed by the N-terminal arginine and lysine. This will act on the peptide bond formed by the N-terminal phenylalanine, tyrosine, tryptophan mainly. Elastase will act on the peptide bond which is formed by the glycine, alanine and serine, and carboxypeptidase will act on carboxyl terminal amino acid, so trypsin, chymotrypsin, elastase and carboxypeptidase will act, and this will convert the polypeptide into oligopeptide, small peptide, peptide, dipeptide, tripeptide, and also the carboxypeptidase will remove the free amino acids. Also on these oligopeptide, dipeptide, tripeptide, pentapeptides, so on these oligopeptide, the intestinal brush border cell, intestinal brush border enzymes that includes the aminopeptidase which removes the N-terminal amino acid, aminopeptidase, dipeptidase, oligopeptidase will act, and this will convert the protein into amino acid or dipeptide, and this is absorbed amino acid and dipeptide. So larger polypeptide, it is broken down by the action of the pepsin into smaller peptide. Then pancreatic enzyme, there is role of pancreatic enzyme. The cholecystokinin and secretin from the intestine causes the release of pancreatic enzymes. So enterokinase, there is release of enterokinase causes the activation of trypsinogen into trypsin. Then trypsin catalyzes the conversion of chymotrypsinogen to chymotrypsin, proelastase to elastase, procarboxypeptidase to carboxypeptidase. Activated enzymes these are the endopeptidase, will cleave the internal peptide bond. This is exopeptidase, carboxypeptidase. Then intestinal brush border enzyme, that is aminopeptidase, dipeptidase and oligopeptidase will act and will convert the peptide into amino acid or dipeptide. Now this is absorbed from the intestine. How the absorption occurs? Three important modes in the absorption of amino acid. The first one is the sodium dependent symport. Most important is the sodium dependent symport. सोडियम डिपेंडेंट सिम्पोर्ट, जैसे ग्लूकोज एब्सॉर्ब होता है, सोडियम डिपेंडेंट ग्लूकोज एब्सॉर्प्शन इज देयर, लाइक दैट द अमाइनो एसिड इज एब्सॉर्ब along with the sodium. So this is intestine, this is the transporter, this is symport, where sodium along with the sodium amino acids, they are internalized. Sodium amino acid transport, symport. So this is secondary active transport, sodium dependent symport. And second one is the transporters, sodium independent transporters are there, which is responsible for transporters, sodium independent transporters are there. So it is the transporters, there is transport for the basic amino acid, there is transporter for the acidic amino acid, there is transporter for glycine and the proline. Then there is transporter for the neutral amino acid. So these transporters exist, which is sodium independent transport into the intestinal cell. And third one is the Mr. cycle, that is gamma glutamyl cycle. But this cycle is very costly. Mr. cycle very rarely used by the renal tissues. It is used by the intestine, renal tissues and brain. It is used Mr. cycle. 3 ATPs, they are consumed for the transport of one single amino acid. It is utilized for the transport of cysteine and glutamine. Cysteine and glutamine, this cycle is utilized. It utilizes 3 ATP for the transport of single amino acid. So it is costly cycle. It is not used routinely, but it is sometimes it is in the intestine, brain, and the renal tissue. It is used. Mr. cycle. So absorption in the intestine. See, sodium dependent import. This is the major mode. Then separate transporters exist for the neutral amino acid, basic amino acid, acidic amino acid, glycine, and amino acid. There are specific transporter which are sodium independent and the Mr. cycle. There are various diseases, that is immunoglycine, celiac disease, then the heart nerves disease, then the pancreatitis. These are the diseases related with the digestion and absorption of the protein. So this is all about the digestion and absorption of the protein. Important short note, SAQ. Now moving to the next important question, that is the next important question, that is urea cycle, ammonia metabolism and urea cycle. So we'll discuss the ammonia metabolism and urea cycle. So how many steps are there? 5 steps are there. Kisne describe ki cycle? Kurt Henslett, Sir Hans Krebs and Kurt Henslett. So it is known as the uh, Krebs Henslett cycle. It is the bicycle. It is like a bicycle जिसका एक चक्का जो है वो Krebs cycle है, दूसरा चक्का जो है वो यूरिया cycle है. So they are connected at different points. So it is also known as the bicycle. It is known as the bicycle. 5 enzymes जिसमें के पहले 2 enzymes है जो माइटोकॉन्ड्रियल है और बाद जिसका नाम A से चालू होता है, बाद में के 3 enzymes है वो साइटोसोलिक है. So it is important cycle. It is important. Ammonia metabolism. So question is ammonia metabolism, LAQ. How ammonia is metabolized in our body? What are the steps of the urea cycle? Mention the urea cycle disorders. Now see, a 9-month-old infant developed poor feeding, hepatosplenomegaly, and convulsions. He has dry and brittle hair. This is a classical finding in one urea cycle disorder. On examination, blood shows increased level of arginosuccinic acid. See, arginosuccinic acid and ammonia. So if there is increased level of ammonia, think about the urea cycle disorder. Think about the urea cycle disorder. In all these 5 types, there is increase in the ammonia level occurs. So what is your probable diagnosis? So these two, they are very severe form: hyperammonemia type 1, hyperammonemia type 2. Okay. If there is citrulline and ammonia, it is citrullinemia. Here there is increase in the arginosuccinic acid and there is increase in the ammonia level, so it is a arginosuccinic aciduria. Along with this classical feature, dry and brittle hair, that is known as trichorexis nodosa. That is known as trichorexis nodosa. See, second, a 30-year-old male infant. 30-year-old male infant कहाँ से आ गया? So 30-year-old male with history of lethargy, poor feeding, and seizure brought to the hospital. Blood shows increased level of citrulline. See here, citrulline and ammonia. Your probable diagnosis is here citrullinemia. We'll discuss this. Just wait. Now see, urea cycle. तो यूरिया साइकिल कहाँ पे होती है? कहाँ पे होती है? लीवर में होती है. पार्टली माइटोकॉन्ड्रियल, पार्टली साइटोसोलिक. कितने स्टेप्स है? 5 स्टेप्स है. इंपॉर्टेंट. कितने इंपॉर्टेंट एंजाइम्स है? 5 एंजाइम्स है. एंजाइम्स कितने हैं? 5. पहली 2 स्टेप्स कहाँ पे होती? माइटोकॉन्ड्रिया में. नेक्स्ट 3 स्टेप्स कहाँ पे होती? साइटोसोल में. नेक्स्ट 3 स्टेप्स कहाँ पे होती? साइटोसोल में. ओके. स्टार्टिंग मॉलिक्यूल क्या है? CO2 एंड अमोनिया. एंड प्रोडक्ट क्या रहेगा? यूरिया. एंड प्रोडक्ट क्या रहेगा? यूरिया. बिफोर द यूरिया साइकिल एक पार्ट हमारा छूट गया, दैट इज अमोनिया मेटाबॉलिज्म. जस्ट वेट. पहले अमोनिया मेटाबॉलिज्म डिस्कस करना पड़ेगा, फिर यूरिया साइकिल पे आएंगे, क्योंकि अमोनिया मेटाबॉलिज्म उतना ही इंपॉर्टेंट है. उसमें ट्रांसअमाइनेशन है, ऑक्सीडेटिव डीअमाइनेशन है, वो छूट जाएगा हमारा, तो वो पहले डिस्कस करेंगे, फिर यूरिया साइकिल पे आएंगे. जस्ट वेट. नाउ सी, ammonia metabolism. So here important, what is important? Sources, transport and toxicity. Sources, transport and toxicity of ammonia is important in the ammonia metabolism. Sources, transport and toxicity of ammonia. Now in the sources, see what are the sources for the ammonia. It includes the transamination, deamination, transamination and deamination. Transamination and deamination first. Second, from the biogenic amines. Third, from the purines and pyrimidines, and by the action of the intestinal bacteria, you will get the ammonia. So from the this is the common source, that is transamination and deamination. Then by the from the biogenic amines, from the purines and pyrimidines, and by the action of intestinal bacteria, you will get ammonia by the intestinal bacteria. Now how it is transported? Now see transamination. What do you mean by transamination here? So transamination is the transfer of amino group from an amino acid to keto acid. Here transamination. In the transamination reaction, there is transfer of amino group from the amino acid to keto acid to form a new pair of amino acid and keto acid. Now see transamination reaction, for example, SGOT. Here the aspartate plus alpha ketoglutarate, it will form the glutamate and oxaloacetic acid. Glutamate and oxaloacetic acid. This reaction, it is catalyzed by the SGOT. Another name is AST, and the pyridoxal phosphate is the coenzyme for this process. Now here all amino acids, they donates their amino group and it is concentrated into a glutamate. Glutamate is the central acceptor molecule for the amino group. So all amino group, they are funneled into glutamate, and glutamate undergoes oxidative deamination in the mitochondria of liver to liberate the ammonia in the oxidative deamination process. So this is how the ammonia is formed. It is concentrated into a glutamate through the various amino acids. Same is for the SGPT, where alanine is the substrate, alpha ketoglutarate in presence of SGPT, where pyridoxal phosphate is the coenzyme. It is alanine transaminase, so it will form the pyruvate plus glutamate. So glutamate is the central acceptor molecule. It accepts the amino group from the most of the amino acid, and it goes to the liver in the form of the glutamine. It goes to the liver, where it is converted into glutamate is converted by the glutamate dehydrogenase. It is oxidative deamination process. It is converted into, or it liberates the ammonia. This is only the transfer of ammonia, transamination, by the process of oxidative deamination. So transdeamination is the process which provides the ammonia for the urea cycle. Now the deamination, see glutamate, it is by the glutamate dehydrogenase reaction. NAD here is converted into NADH. Glutamate dehydrogenase reaction, it is converted into alpha ketoglutarate and ammonium ions, and it is ammonium ions. It is liberated by the oxidative deamination process. Also D-amino acid oxidase, L-amino oxidase are there, which liberates the ammonia level. Also some non oxidative deamination processes are there, D-sulfhydrase, histidase. This also causes the release of ammonia. Okay, so transamination and oxidative deamination process which provides the ammonia. And in how it is transported, so ammonia, it is transported in the form of glutamine. It is transported by in the form of glutamine from brain and intestine, from brain and intestine to liver in the form of glutamine, while in the form of alanine by the glucose alanine cycle, it is from the muscle to the liver, in the form of alanine. So this is transport form of the ammonia in the form of glutamine from the brain and intestine to the liver, and in the form of alanine from muscle to the liver. Now, why ammonia is toxic to the brain? Why it is toxic to the brain? Because increase in the ammonium ion, or increase in the ammonia level, it combines with the alpha ketoglutarate of the TCA cycle, and it forms glutamate. This glutamate, again it is converted into glutamine by accepting the NH3. It is converted into glutamine. So glutamine concentration increases in the central nervous system, and it causes the edema, cerebral edema. And decrease in the alpha ketoglutarate level, it hampers the TCA cycle. It decreases the TCA cycle, decreases the ATP production. So it decreases the ATP production in the brain, which is purely dependent on the oxidative process for the ATP production. So it causes the toxicity. So increase ammonia level combines with the alpha ketoglutarate, hampers the TCA cycle, decreases the ATP production in the brain. That's why it is toxic. Increase ammonia level, it is toxic. Also increase glutamine concentration in the brain causes the cerebral edema. Decrease in the glutamate also decreases the GABA production, gamma amino butyric acid production. This is also a reason. So this is important reason why ammonia is toxic to the brain. So we discussed the transport, means we discussed the synthesis of ammonia. It is by the transamination and oxidative deamination process, or non-oxidative deamination process. There is liberation of ammonia occurs from the biogenic amines. You will get the ammonia from the purine and pyrimidine, and by the urea splitting organism, intestinal bacteria, which converts the urea into ammonia. So these are the sources of ammonia. It is transported in the form of glutamine and alanine. Then it is toxic because it depletes the alpha ketoglutarate level of the TCA cycle. That's why it is toxic. And now how it is disposed of? So ammonia is converted into urea and it is disposed of. So see, urea cycle, 5 enzyme, 5 steps. First 2 steps occurs in the mitochondria. Next 3 steps occurs in the cytosol. It occurs in the liver. It is also known as ornithine cycle. It is known as ornithine cycle. It is occurs in the liver because of one important enzyme, that is arginase. Arginase enzyme, it is only present in the liver. That's why urea cycle occurs in the liver. Now see the steps of the urea cycle. See, so it is CO2 plus ammonium ions combine. CO2 plus ammonium ion, it forms carbamyl phosphate. Here enzyme is carbamyl phosphate synthetase 1. 2 molecules of ATP utilized here, which is converted into 2 ADP plus inorganic phosphate. And N-acetyl glutamate is the allosteric activator of this carbamyl phosphate synthetase 1. Okay, so carbamyl phosphate synthetase 1. This is mitochondrial enzyme. Now this carbamyl phosphate, it is combines with the ornithine. To form citrulline. And the name of the enzyme is ornithine transcarbamylase. and it is present in the mitochondria. It is present in the mitochondria. So first step, first first two steps, they occurs in the mitochondria. So ornithine, it is converted into citrulline. Now citrulline condenses with the aspartate. It condenses with the aspartate, and ATP in this reaction, it is converted into AMP and inorganic phosphate. AMP and inorganic phosphate. So two, it is equal to the two phosphate bonds, two high energy phosphate bonds. They are utilized here. So citrulline condenses with aspartate to form the arginosuccinic acid. Arginosuccinic acid. Then arginosuccinate lyase. Arginosuccinate lyase will act, will liberate the fumarate and will form the arginine. Arginine, then it forms the urea. Arginine, it forms the urea by the enzyme arginase. Now see, just wait. So this is arginase, this is arginosuccinate lyase, and this is arginosuccinate synthetase. Okay, so this is third, this is fourth, this is fifth. So first two enzymes, they are present in the mitochondria, जिनका नाम ए से चालू होता, वो सब साइटोसोल में रहते हैं. आर्जिनोसक्सिनेट सिंथेटेज, आर्जिनोसक्सिनेट लाइस एंड आर्जिनेज, दे आर साइटोसोलिक एंजाइम. यहाँ पे ऑर्निथिन आने के बाद ऑर्निथिन को साइटोसोल से माइटोकॉन्ड्रिया में ट्रांसपोर्ट करना पड़ता. उसके लिए जो ट्रांसपोर्टर लगता, उसका नाम है ऑर्निथिन ट्रांसपोर्टर. And it is encoded by the protein, that is ORNT1 gene. Okay, so five enzymes, five steps. Activator is N-acetyl glutamate. Rate limiting enzyme is carbamyl phosphate synthetase 1. So carbamyl phosphate synthetase 2 is involved in the pyrimidine synthesis. 2 is involved in the pyrimidine synthesis, which is cytosolic enzyme. This is mitochondrial enzyme. So CO2 plus NH4 combine to form carbamyl phosphate, which condenses with ornithine in presence of ornithine transcarbamylase to form the citrulline. Citrulline condenses with aspartate with the help of arginosuccinate synthetase, and there is utilization of two high energy phosphate bonds, which is equal to the 2 ATP utilization. It is converted into arginosuccinic acid. Arginosuccinic acid, by the lyase reaction, there is release of fumarate. And it is converted into arginine. Arginine is acted upon by the arginase, and hydrolysis reaction, there is formation of the urea. So one nitrogen group of urea, it is from the ammonia molecule, and one is from the aspartate molecule. So C NH2, this is NH2. So one is from the aspartate, one is from the ammonia. This is urea. So these are the steps of the urea cycle. Regulation, it is by the compartmentalization, and second one is the CPS one, where N-acetyl glutamate is the allosteric activator. See, so how urea cycle, it is linked with the Krebs cycle. That's why it is known as the Krebs bicycle. It is bicycle. Now see, the oxaloacetate, which is in the TCA cycle, it is transaminated into aspartate. Oxaloacetate, it is transaminated into aspartate, and aspartate, there is formation of arginosuccinate, and by the lyase reaction there is release of fumarate. So fumarate enters the this TCA cycle. So there is connection between oxaloacetate and aspartate, and fumarate, it is connected with the TCA cycle. There is production of ATPs in the form of NADH, or there is direct formation of ATP in the TCA cycle, and ATPs, they are utilized in the first and third step. it is utilized in the first and third step. ATP, it is utilized here. So here production in the TCA cycle, it is utilized here. Okay. Then CO2 it is produced in the TCA cycle, and it is a starting product in the urea cycle. Starting product in the urea cycle. This is how they are connected with each other. Connected with each other. So see CPS1 and CPS2 Viva question. So CPS1 carbamyl phosphate synthetase 1, carbamyl phosphate synthetase 2. These are the examples of biotin independent carboxylation reaction. For this carboxylation reaction, biotin does not require. So CPS1 carbamyl phosphate synthetase 1, it is present in the mitochondria. CPS2, it is present in the cytosol. This is urea synthesis. This is pyrimidine nucleotide synthesis. Here source of nitrogen is ammonia. Here source of nitrogen is glutamine. N-acetyl glutamate is the allosteric activator here, and it is CPS2. Generally, it is activated by PRPP, phosphoribosyl pyrophosphate. So it is no inhibition for the CPS1. It is cytidine triphosphate, which is the end product of this pathway, which inhibits the CPS2. So this is a Viva question. Enumerate the difference between CPS1 and CPS2. These are the different urea cycle disorders. So there are 5 disorders and one hyperornithinemia, means ornithine बढ़ जाएगा because of ORNT1 gene mutation, the transporter defect. So 5 एंजाइम के साथ 5 रिलेटेड है और एक ट्रांसपोर्टर डिफेक्ट, ऐसे 5 डिसऑर्डर्स है. तो पहले दो जो है हाइपरअमोनिमिया टाइप 1 एंड टाइप 2, दे आर सीवियर फॉर्म, they are fatal, they are severe form. Their ammonia level, where ammonia level increases very high, ammonia level increases very high in the first 2 disorders, that is hyperammonemia type 1 and hyperammonemia type 2. There is severe increase in the ammonia level. So this is due to CPS1 defect. This is ornithine transcarbamylase defect. Along with this, there is orotic aciduria in this second hyperammonemia type 2. There is orotic aciduria. Okay, and this is X-linked dominant, partially dominant hyperammonemia type 2. Then this is HHH syndrome, that is hyperornithinemia. It is due to ORNT1 mutation. ORNT1 mutation, transporter defect, ornithine transporter defect. There is increased ornithine, so hyperornithinemia, hyperammonemia, and homocystinuria. These are the 3 classical features of this hyperornithinemia. Then citrullinemia, there is increased citrulline level, there is increased citrulline level and the ammonia level, citrulline and ammonia level. It is due to arginosuccinate synthetase defect. Then arginosuccinic acid urea, it is due to arginosuccinate lyase defect, and there is increased arginosuccinate level and trichorexis nodosa. There is also increased ammonia level, but these are less severe because ammonia level, as we go on, अमोनिया लेवल मतलब ये शुरू के इसमें अमोनिया ज़्यादा बड़ा हुआ रहता, बाद में थोड़ा कम बड़ा हुआ रहता, तो लेस सीवियर रहता. सो द arginosuccinate aciduria, there is arginosuccinate level and trichorexis nodosa, friable tufted hair, easily pluckable hair. Trichorexis nodosa is a feature of arginosuccinic aciduria. And hyperargininemia, because of arginase defect, there is increase in the arginine level, there is increase in the ammonia level in hyperargininemia. तो 5 एंजाइम 5 डिफेक्ट. टाइप 2 जो है उसमें ओरोटिक एसिड्यूरिया रहता और आर्जिनोसक्सिनिक एसिड्यूरिया है, इसमें ट्राइकोरेक्सिस नोडोजा, फ्राइबल टफ्टेड इजीली प्लकेबल हेयर्स. इट इज ट्राइकोरेक्सिस नोडोजा, इट इज ए फीचर ऑफ आर्जिनोसक्सिनिक एसिड्यूरिया. और उसमें ये क्यों रहता, इसके डिफेक्ट में दिस इज एक्स लिंक पार्शियली डोमिनेंट. सो ऑर्निथिन ट्रांसकार्बोमाइलेज डिफेक्ट, देर इज orotic aciduria because in the block, if there is block here, this carbamyl phosphate, it is diverted for the pyrimidine nucleotide pathway, and orotic acid is the intermediate of pyrimidine nucleotide synthesis pathway. That's why there is increased orotic acid level in the ornithine transcarbamylase defect. Okay, So this is about the ammonia metabolism, urea cycle, regulation of urea cycle, link between urea cycle and TCA cycle, difference between CPS1 and CPS2, and various urea cycle disorders. So this is very, very important LAQ long answer question. Very, very important long answer question. Hello everyone. Now the topic of discussion is phenylalanine and tyrosine metabolism. Hi Vinay. Hi Poco. Hi Pratham. Poco Pratham Asia. Hello. Hi Snehajit. Still listening. Hello everyone. This is the revision topic. So just we completed the ammonia metabolism and urea cycle. So this is important. You may get a case on the urea cycle disorder that may be on the hyperammonemia type 1 or hypoammonemia type 2. You may get a case. अगर आपको डिफरेंशिएट करना नहीं है और अमोनिया बढ़ा हुआ दिया है तो जस्ट राइट डाउन यूरिया साइकिल डिसऑर्डर. जस्ट राइट डाउन यूरिया साइकिल डिसऑर्डर. ओके. कंप्लीट मार्क तो नहीं मिलेंगे लेकिन मार्क मिलेंगे. ओके. इफ यू कैन नॉट डिफरेंशिएट बिटवीन दिस 5. सो ऐसा स्पेसिफिक तो जनरली नहीं आएगा, बट इट कैन बी ए लॉन्ग ऐसे टाइप क्वेश्चन ऑन द यूरिया साइकिल डिसऑर्डर. ओके. सो जस्ट वेट 2 मिनट, विल कवर द फेनिलैलानिन टायरोसिन कवर करते. मेरे पानी पीने दो थोड़ा सा. आदि. हाय जोसेफ. हाँ सभी कवर कर लेंगे. ज़्यादा टाइम नहीं लगता. नाउ दिस इज इजी टॉपिक. फेनिलैलानिन टायरोसिन ट्रिप्टोफैन. सी. ammonia metabolism and the urea cycle, this is very very important. it is मतलब जैसे कार्बोहाइड्रेट मेटाबॉलिज्म में ब्लड ग्लूकोज रेगुलेशन है, वैसे ही फेनिलैलान वैसे ही प्रोटीन मेटाबॉलिज्म में यूरिया साइकिल और उसके डिसऑर्डर्स है. ठीक है. गैमी प्रो. ब्लैक लवर गर्ल. हेलो एवरीवन. कौन कौन है जो शुरू से सुन रहा है, मतलब जब से स्टार्ट हुआ तब से सुन रहा है? ऐसे कितने लोग हैं? किसी किसी का नेट पैक खत्म हो गया होगा, है ना? स्नेहाजीत, हाय प्रथम, विनय। समझा क्या? कुछ कुछ रिवाइज हुआ क्या? कुछ फायदा हुआ क्या? चलो तो चालू करते फेनिलएलैनिन एंड टायरोसिन मेटाबॉलिज्म। दिस इज इंपॉर्टेंट टॉपिक। थैंक यू एवरीवन, थैंक यू एवरीवन। तो विल स्टार्ट विद द फेनिलएलैनिन एंड टायरोसिन मेटाबॉलिज्म। मैंने पहले भी बोला है, जब भी इंडिविजुअल अमीनो एसिड मेटाबॉलिज्म पे क्वेश्चन आता है, जस्ट ट्राई टू फर्स्ट राइट डाउन द प्रॉपर्टीज ऑफ दिस अमीनो एसिड, देन राइट डाउन द फ्लो चार्ट एंड देन गो फॉर द pathway. Okay, pathway proper. So here see, phenylalanine, aromatic amino acid, both aromatic amino acid, phenylalanine, tyrosine, aromatic amino acid. Then they are non-polar, non-polar, both they are non-polar. Then phenylalanine is the essential amino acid. This is non-essential amino acid, non-essential amino acid. Aromatic amino acid belongs to non-polar. This is phenylalanine essential, tyrosine non-essential amino acid. In the metabolic classification, they will give both glucose and ketone, means glucose and fat. So they are glucogenic and ketogenic both. They are both glucogenic and ketogenic. Phenylalanine, tyrosine. Tyrosine having the phenolic hydroxyl group, it is having the benzene ring. Phenylalanine having the benzene ring. Now in the metabolism, see, phenylalanine, it is converted into tyrosine. Tyrosine will give you glucose. Tyrosine will give you fat, and tyrosine, it will give you dopamine in the central nervous system. It will give you epinephrine, norepinephrine in the adrenal medulla. Then it will give you melanin in the skin, hair, and eyes, and it will give you thyroid hormone T3 and T4 in the thyroid gland. So this is overview of phenylalanine and tyrosine metabolism. Apart from this, it is also incorporated during the protein synthesis. It is incorporated during the protein synthesis. So this is important. Now see, phenylalanine is converted into tyrosine. Tyrosine will give both glucose and fat. Tyrosine is incorporated during the protein synthesis. These are the specialized product given by the phenylalanine, tyrosine: dopamine, epinephrine, norepinephrine, melanin, thyroid hormone T3 and T4. T3 and T4. Now, in the metabolism, see how it is converted into glucose and fat. It is important how tyrosine will give you glucose and fat. This is important in the metabolism, and various inborn errors, they are related with phenylalanine and tyrosine metabolism. And you may get short note on the specialized product given by the phenylalanine and tyrosine. Now see the first reaction, which is important, in which phenylalanine, it is converted into tyrosine. It is tyrosine, nothing but the para-hydroxy phenylalanine. Tyrosine, phenylalanine is converted into tyrosine. So tyrosine is nothing but the para-hydroxy phenylalanine. In this process, one atom of oxygen is incorporated into a substrate and another is reduced to form water molecule. And this reaction is tetrahydrobiopterin dependent. In this reaction, tetrahydrobiopterin is converted into dihydrobiopterin, and with the help of dihydrobiopterin reductase, NADPH dependent dihydrobiopterin reductase, this dihydrobiopterin is converted into tetrahydrobiopterin. For this reaction, the important enzyme is phenylalanine hydroxylase. And deficiency of this causes the phenylketonuria, in which phenylalanine is diverted for the production of various products: phenyl pyruvate, phenyl acetate, phenyl lactate, phenyl acetyl glutamine. These are the different products which are produced in the absence of phenylalanine hydroxylase, that is known as phenylketonuria. Now this the further pathway is, see, This tyrosine, it is converted into, it is transamination process. So tyrosine, it is converted into para hydroxyphenyl. So this is para hydroxyphenylalanine. This is para hydroxyphenyl pyruvate. Para hydroxyphenyl pyruvate. And this para hydroxyphenyl pyruvate, it is converted into homogentisate. Homogentisate, which is acted upon by the homogentisate oxidase, and it is converted into 4-methyl acetoacetate. Acetoacetate. This 4-methyl acetoacetate is isomerized into 4-fumaryl acetoacetate, and which will give you fumarate and acetoacetate. Fumarate and acetoacetate. So fumarate will give you glucose, acetoacetate will give you fat. So enzymes, now see, the tyrosine by the transamination process, this is tyrosine transaminase. Tyrosine transaminase, here the alpha ketoglutarate is converted into glutamate. So by the process of transamination, there is transfer of amino group from the tyrosine to the alpha ketoglutarate and conversion of alpha ketoglutarate into a glutamate. This is how amino group is transferred to the glutamate. Now this is by the transaminase, tyrosine transaminase, transamination, pyridoxal phosphate dependent reaction. Tyrosine is converted into para-hydroxyphenyl pyruvate. Now para-hydroxyphenyl pyruvate hydroxylase, this is vitamin C dependent reaction. Vitamin C is required here. So hydroxylase, para-hydroxyphenyl pyruvate is converted into homogentisate. Homogentisate is nothing but the para-hydroxyphenyl acetate. Now see here, phenylalanine, it is converted into para-hydroxyphenylalanine. Para-hydroxyphenylalanine is converted into para-hydroxyphenyl pyruvate by the transamination reaction, and para-hydroxyphenyl pyruvate is converted into para-hydroxyphenyl acetate, that is homogentisate. Now homogentisate is converted into 4-methyl acetoacetate, 4-fumaryl acetoacetate, which is hydrolyzed by the hydrolase. See, it is homogentisate oxidase. Gentisate oxidase. For 4-methyl acetoacetate, this is isomerase, and this is fumaryl acetoacetate hydrolase. Hydrolase will split this into fumarate and acetoacetate. So this is how the tyrosine and phenylalanine, they are both glucogenic and ketogenic. The fumarate will give you the glucose and acetoacetate will give you the fat. Now the blockade in these steps, various steps, will give the different inborn errors of metabolism. So see, all of you know that this deficiency of this block in this, it causes the phenylketonuria. Phenylketonuria. So why it is known as phenylketonuria? Because there is excretion of phenyl pyruvate, which is a keto acid. That's why it is known as phenylketonuria. Why there is mousy odor of urine? Because of excretion of phenyl acetate and other byproducts in the urine. Be diplomatic here. Phenyl acetate and other product, क्योंकि कुछ कुछ बुक्स में phenyl lactate भी दिया है. सो रेफरेंस बुक में फेनाइल एसिटेट ही दिया है, बट बी डिप्लोमेटिक हियर. एक्सक्रीशन ऑफ फेनाइल एसिटेट एंड अदर बाय प्रोडक्ट्स विल गिव माउसी ऑर्डर टू द यूरिन. So this phenylalanine is diverted for the production. It is converted into phenyl pyruvate. Phenyl pyruvate. This is converted into phenyl acetate. This is converted into phenyl lactate. And this is converted into phenyl acetyl glutamine. So this is keto acids, they are excreted in urine, that's why it is phenylketonuria. Mousy odor is because of phenyl acetate excretion in the urine. Now in this disease there is mental retardation, neurological manifestation, seizures, tremors, mental retardation is there. This is because of decreased production of neurotransmitters, dopamine, norepinephrine, epinephrine, because of uh, block in this pathway, decreased synthesis of neurotransmitters. Also there is, it hampers the tryptophan metabolism, and there is decreased synthesis of serotonin. That's why neurological manifestations are there in the this phenylketonuria. Apart from the neurological manifestation, the phenylalanine is important for the tyrosine synthesis. So tyrosinase is the only enzyme which is inhibited by increase in the phenylalanine concentration. That's why there is hypopigmented patches. So hypopigmented patches all over the body. So there is decreased pigmentation of eyes, decreased pigmentation of hair, and the hypopigmented patches all over the body. phenylketonuria, blue eyes रहती, blue eyes, phenylketonuria. जरूरी नहीं है हर केस में रहेगा वैसा. Okay, and it is the screening test is the Guthrie test, which is an bacterial inhibition assay done by using the Bacillus subtilis growth. So Guthrie test is the screening test for the diagnosis of phenylketonuria, while the this tandem mass spectrometry, it is the confirmatory test for the diagnosis of phenylketonuria. So this is important case, phenylketonuria. Now tyrosine transaminase, there are two T's, tyrosine transaminase. So it is tyrosinemia type 2. Tyrosinemia type 2. It is also known as the Richner-Hanhart syndrome. There is oculocutaneous manifestation, there is rash, and there is ocular lesion in this disease, that is tyrosinemia type 2. Deficiency of para-hydroxyphenylpyruvate hydroxylase causes neonatal tyrosinemia. Neonatal tyrosinemia. Homogentisate oxidase, it is important. The deficiency causes Alkaptonuria. Alkaptonuria. Deficiency of homogentisate oxidase causes the Alkaptonuria. There is accumulation of homogentisate which is diverted for the benzoquinone acetate formation, and benzoquinone acetate on polymerization from the alkaptone, which is deposited in the various joints, in the tendons, in the cartilage, in the skin, and that gives black pigmentation to that tissues. So black cartilage and the deposition of alkaptone into the joints causes the uchronosis. Uchronosis, it is the condition where alkaptone pigment deposition occurs in the joints. So long standing alkaptonuria means after the age of 30 to 40 years there is tendency to develop the arthritis, and symptomatic treatment is needed for the arthritis. As such, no treatment is needed. Only classical feature is excretion of urine which turns dark on standing on exposure to air. Because of formation of pigment, black pigment, it turns dark on standing. Means this homogentisic acid, it is excreted, homogentisate, it is converted into benzoquinone acetate, and then alkaptone, that black pigment is there. So it is black urine on standing. This is a feature. As such, individual is asymptomatic, but tendency to develop arthritis in the later ages. So black diaper, black staining of the diaper will be there in the alkaptonuria. Then the fumaryl acetoacetate hydrolase deficiency, it causes the tyrosinemia type 1. Mainly this deficiency causes the tyrosinemia type 1. So this is the fatal condition. It involves the liver and the kidneys. It is fatal, tyrosinemia type 1, and there is cabbage like order of the body secretions. Cabbage like order, tyrosinemia type 1. Cabbage like order of the body secretion. So tyrosinemia type 2 due to tyrosine transaminase defect, para-hydroxyphenylpyruvate hydroxylase defect, neonatal tyrosinemia, homogentisate oxidase causes the alkaptonuria, and the fumaryl acetoacetate hydrolase defect causes the tyrosinemia type 1. And then the specialized product, what are the different specialized product given by the phenylalanine and tyrosine? So it will give you the neurotransmitters. For example, see tyrosine, it is converted into dopa. Dopa is converted into dopamine. Dopamine hydroxylase convert dopamine into norepinephrine. Norepinephrine, and norepinephrine, it is converted into epinephrine. So this is catecholamine synthesis. This is catecholamine synthesis. Here it is tyrosine hydroxylase hydroxylase, tyrosine hydroxylase. This is decarboxylation reaction, which is pyridoxal phosphate dependent. Decarboxylase, decarboxylase. Then dopamine hydroxylase, and this is SAM, S-adenosyl methionine, which donates the methyl group here, transmethylation reaction, where remember norepinephrine, no methyl group, it is converted into epinephrine. Methyl transferase reaction where SAM donates the methyl group, S-adenosyl methionine donates the methyl group. So this is how the dopamine, norepinephrine and epinephrine synthesis means catecholamine synthesis. Catecholamines synthesis. Catecholamine synthesis from the tyrosine, specialized product. So pheochromocytoma is a disease, it is a adrenal tumor, pheochromocytoma, adrenal gland tumor where there is increased synthesis of catecholamines occurs. Pheochromocytoma, adrenal gland tumor where there is increased production of epinephrine, norepinephrine occurs, and there is increased excretion of vanillyl mandelic acid in urine, in the urine occurs. So it is characterized by raised blood pressure, severe headache, palpitation. So these are the symptoms of pheochromocytoma, adrenal gland tumor, catecholamine synthesis, phenylalanine and tyrosine metabolism. Again, one more disease which is connected with this, that is the Parkinson's disease. Parkinson's disease. Wait, Parkinson's disease. In Parkinson's disease, there is decreased production of dopamine in brain because of degeneration. Parkinson's disease, this affects near about the 1% people above the age of 60 years. This is due to the degeneration of certain parts of the brain, that is basal ganglion, substantia nigra and locus coeruleus of the brain. So there is decreased dopamine production in the Parkinson's disease, which is characterized by rigidity. In the movements there is rigidity, there is tremors, involuntary movements, mask like face, mask face, like face. So these are the features of Parkinson's disease. In this, the treatment dopa is given, dopamine is not given because dopamine does not crosses the blood brain barrier. So dopa is given, L-dopa along with the carbidopa is given in the treatment. So L-dopa crosses the blood brain barrier, enters in the brain, which is converted into dopamine and act, and peripheral conversion of dopa to dopamine is prevented by the carbidopa, which is given with the dopa in the treatment of the disease. So Parkinson's disease, rigidity, tremor, involuntary movements, mask like face, these are the features of Parkinson's disease due to degeneration of certain parts of the brain, basal ganglion, degeneration of certain parts of the brain above the age of 60 years. Parkinson's disease above the age of 60 years, 1% population above the age of 60 years. So this is pheochromocytoma, this is Parkinson's disease. Okay, then the tyrosine, it is important for the thyroid synthesis of T3 and T4 in the thyroid gland. There are 4 basic steps in the thyroid hormone synthesis, that is activation of iodine, then attachment of iodine to tyrosine residue, of thyroglobulin, then coupling reaction and the proteolysis. So these are the 4 steps. So this is thyroglobulin residue, this is tyrosine of the thyroglobulin residue. To this tyrosine there is attachment of iodine occurs, and in the coupling this monoiodotyrosine combines with the diiodotyrosine. So monoiodotyrosine combines with the diiodotyrosine to form triiodotyrosine, that is T3, and the diiodotyrosine combines with the diiodotyrosine to form the T4. So this is how the thyroid hormone synthesis occurs from the tyrosine. thyroid hormone synthesis occurs from the tyrosine. And the fourth important product is, that is the melanin synthesis. The only enzyme in the melanin synthesis is tyrosinase. So in the tyrosinase, see, in the melanin synthesis, this tyrosine, by the tyrosinase, it is converted into dopa. Dopa is converted into dopaquinone, and through various steps, this dopaquinone, it is converted into melanin. That is 5,6 dihydroxy indole 5,6 quinone, and this is this forms the melanin. So this is tyrosinase is only involved in the tyrosinase is the only enzyme involved in the melanin synthesis. Tyrosinase. Okay, so albinism is the disorder in which there is tyrosinase is defective and decrease in the melanin synthesis occurs. So there is generalized hypopigmentation in the tyrosinase defect. So that is known as albinism. So there is decreased pigmentation of hair, eyes and skin, decreased pigmentation. There is generalized hypopigmentation all over the body. So albinism. So generalized hypopigmentation all over the body, pigmentation all over the body. Then the photophobia and nystagmus, to and fro movements of the eyeball. Nystagmus, these are the features of the albinism. फोटोफोबिया, निस्टैग्मस एंड द हाइपोपिगमेंटेशन. तो उसको ट्रीटमेंट में क्या देंगे? सन ग्लासेस देंगे उसको, सनस्क्रीन लोशन लगाने को बोलेंगे, हाई क्या बोलते हैं उसको, पीपीएफ नहीं, पीपीएफ नहीं, क्या बोलते हैं उसको, एसपीएफ, है ना. तो वो सन प्रोटेक्शन के लिए उसको क्रीम लगाने को बोलेंगे और उसको आई ग्लासेस वगैरह देंगे. सन एक्सपोजर अवॉइड करने को बोलेंगे, बिकॉज दिस इज the ultraviolet rays can be carcinogenic in this patient. So albinism, it is related with the melanin synthesis. So this is all about the phenylalanine and tyrosine metabolism, specialized product given by the phenylalanine tyrosine, and how it is glucogenic and ketogenic. So this is all about the phenylalanine and tyrosine metabolism. So black urine in infancy is a diagnostic for, see this MCQ. हाँ, SPF 15, correct है. Snehajeet, L-dopa and carbidopa, it is given in the treatment. So L-dopa and carbidopa, see dopamine देते नहीं क्योंकि blood brain barrier क्रॉस नहीं करता. एल डोपा ब्रेन में जाके डोपामिन में कन्वर्ट होता और वो यूजफुल है वहाँ पे. पेरिफेरल साइड इफेक्ट उसके प्रिवेंट करने के लिए कार्बिडोपा साथ में दिया जाता. वो उसका डोपा को डोपामिन में कन्वर्शन बाकी के टिश्यूज में नहीं करता, तो उसके जो साइड इफेक्ट्स है वो प्रिवेंट किए जाते उससे. सो दैट इज द कॉम्बिनेशन. हाँ, यस. अलोंग विद द, ये तो लिखना पड़ेगा ना, नॉर इपिनेफ्रिन, इपिनेफ्रिन और डोपामिन बन ही नहीं रहा, और ट्रिप्टोफैन का मेटाबॉलिज्म इनिबिट करने की वजह से सीरोटोनिन का प्रोडक्शन नहीं हो रहा. दिस इज द रीजन फॉर मेंटल रिटार्डेशन इन द phenylketonuria. सो अल्कैप्टोन्यूरिया एज सच असिम्टोमेटिक ब्लैक डाइपर रहेगा, या एक्सपोजर जब इसको टाइम लगता है ऑन एक्सपोज टू एयर इट टर्न्स ब्लैक, बट इट इज ऑफन गोज अननोटिस. लेकिन अभी डाइपर के वजह से ना ये आइडेंटिफाई होता है जल्दी, क्योंकि डाइपर हमने सपोज 40 रुपए का डाइपर पहना है बच्चों को, तो उसके पैसे वसूल करने रहते ना हमको, तो वो ज़्यादा देर तक पहना के रखते. तो वो ब्लैक स्टेनिंग ऑफ डाइपर कैन बी यूजफुल in the diagnosis of the alkaptonuria. PKU, mousy odor of urine, mental retardation, Guthrie test, ferric chloride test, MSUD, maple syrup urine disease, burnt sugar, homocystinuria, subluxation of the lens, increase homocysteine level, cyanide nitrous peroxide test positive. So this is the black deposits on the palmar crease, ear cartilage, and the conjunctiva. There is deposition of black pigment in the alkaptonuria. This is the diagnosis. The condition is known as ochronosis. Ochronosis because of alkaptone. deposition of pigment alkaptone. So this is albinism. This is due to tyrosinase defect in the tyrosinase. Albinism, hypopigmented hair, skin is there in the albinism. Now coming to the tryptophan metabolism. Tryptophan metabolism, it is in the tryptophan metabolism there is two important pathway. So पहले tryptophan के बारे में discuss करो. This is aromatic amino acid. This is aromatic amino acid. Then it is bulky amino acid. It contains indole ring is there. Indole ring is there in the tryptophan. It gives in metabolism both glucose and fat. So both glucogenic and ketogenic. So aromatic amino acid, phenylalanine, tyrosine, tryptophan, and the isoleucine, they will give both glucose and fat. So both glucogenic and ketogenic amino acid. So tryptophan, in the overview, tryptophan will incorporate into the protein during the protein synthesis. It will give you glucose and fat both. Glucose and fat both. In the specialized product, it will give you the tryptophan will give you melatonin. It will give you serotonin, melatonin. Then it is converted into NAD and NADP. NAD and NADP. It is tryptophan is converted. 60 milligram of tryptophan will give 1 milligram of niacin. So it is important for the production of NAD, NADP. Tryptophan is converted into serotonin. Tryptophan is converted into melatonin. It will give you glucose. It will give you fat. It is incorporated during the protein synthesis, and it is excreted in the urine in the form of hydroxyindole acetic acid. Excreted in the urine in the form of hydroxyindole acetic acid. So tryptophan metabolism as such, the all steps they are not important here. So remember that tryptophan follows two important pathways. The first one is the kynurenine anthranilate pathway, and second one is the serotonin pathway. Kynurenine anthranilate pathway, and second one is the serotonin pathway. So see, tryptophan in the kynurenine anthranilate pathway, tryptophan is converted into kynurenine. It is converted into anthranilate, and anthranilate it will give you the NAD, NADP. So this is the kynurenine anthranilate pathway. Now there are multiple intermediates in between this. Tryptophan is converted into N-formyl kynurenine. N-formyl kynurenine is converted into kynurenine. Kynurenine is converted into 3-hydroxykynurenine, and 3-hydroxykynurenine is converted into 3-hydroxyanthranilate. And through the various intermediate, it forms the 2-amino 3-carboxy muconate semialdehyde, which forms the quinolinate, and quinolinate is converted into nicotinamide mononucleotide, which is converted into NAD and NADP. So this is overall pathway, kynurenine anthranilate pathway. So there is role of the in B6 deficiency, pyridoxal phosphate deficiency. This 3-hydroxykynurenine, it is C 3-hydroxyanthranilate. Okay, so kynureninase, this is pyridoxal phosphate dependent reaction. B6 is required, pyridoxal phosphate is required for this step. In the B6 deficiency, this kynurenine, in this deficiency, it is converted into xanthurinate and excreted in urine. And it is excreted in the urine in the pyridoxal phosphate deficiency. Kynurenine, it is excreted in the urine. So this, in this step, it is also converted into alanine, that will give you glucose. Alanine, and that will give you glucose. This will also give you acetoacetate, that will give you fat. So it is glucogenic, ketogenic both. Tryptophan, it is glucogenic, ketogenic both. It will give you alanine, that will give you glucose. It will give you through the intermediate, it will give you fat. and it is glucogenic and ketogenic both. In the B6 deficiency, pyridoxal phosphate deficiency, the kynurenine is diverted for the xanthurenate production and it is excreted in the urine in patient with pyridoxal phosphate deficiency. Also maize and jawar eating population, in maize the niacin it is present in the bound form, so it is unavailable for the absorption, while high jawar has the leucine content, high leucine content, which inhibit the QPRTase enzyme which is involved in the this NAD synthesis, and it is inhibited by the high leucine content of the jawar, high leucine content of the jawar. So jawar and maize eating population, there is deficiency of niacin occurs. Jawar and maize eating population, when it is taken as a staple diet, it is taken as a staple diet. So this is kynurenine anthranilate pathway, and in the serotonin pathway, this tryptophan it is converted into 5-hydroxy tryptophan, and this 5-hydroxy tryptophan it is converted into 5-hydroxy tryptamine, that is serotonin. So serotonin it is a cerebral excitatory neurotransmitter. It is important in the central nervous system. It maintains the blood pressure. Serotonin it is cerebral excitator, maintains the blood pressure. So tryptophan is converted into 5-hydroxy tryptophan by the enzyme that is tryptophan hydroxylase, and by the this decarboxylase it is converted into serotonin. Now N acetylation serotonin is converted into N acetyl serotonin, and N acetyl serotonin by the transfer of methyl group by the SAM it is converted into melatonin. So melatonin is the important. Melatonin it is secreted by the pineal gland, and melatonin it decreases the melanocyte stimulating hormone. It decreases the ACTH production, and it is important for the sleep wake cycle. Melatonin it is the natural sleep inducer. It is important for the sleep wake cycle, and melatonin inhibit the ACTH and MSH production in the body. So this is the important pathway, that is serotonin pathway. So this serotonin it is by the monoamine oxidase it is converted into hydroxyindole acetic acid, and it is excreted in the urine. It is excreted in the urine. Okay, so this is serotonin pathway. This is kynurenine anthranilate pathway in the tryptophan metabolism. What are the diseases which are related with the tryptophan metabolism? One is the heart nerves disease. Heart nerves disease. In heart nerves disease, there is mental retardation and there is niacin deficiency manifestations, that is pellagra-like symptoms. Mental retardation, pellagra-like symptoms. Heart nerves disease is due to decreased absorption of tryptophan from the intestine. And there is ataxia-like symptoms. Mental retardation, ataxia means lack of coordination, and the pellagra-like symptoms in the heart nerves disease. Apart from this, carcinoid syndrome. Carcinoid syndrome, it is argentaphinoma. It is argentaphinoma. It is a tumor of argentaphin cell of the intestine where the more than 60% tryptophan it is diverted for the serotonin production. So increased serotonin causes the symptoms. It causes the diarrhea, facial flushing, increased heart rate. These are the symptoms of due to increased serotonin production. This is carcinoid syndrome or argentaphinoma uh, uh, where more than 60% of tryptophan is diverted for the serotonin production. So that causes the symptoms, carcinoid syndrome or argentaphinoma. So heart nerves disease, carcinoid syndrome. Then vitamin B6 deficiency, there is xanthurenate excretion in the urine, and when we take jawar as a staple diet, or leucine jawar as a staple diet, it inhibits the QPRTase enzyme, it causes the niacin deficiency. So these are the different diseases related with the tryptophan metabolism. See, this is the golden image. बहुत सारे एंट्रेंस एग्जाम के क्वेश्चन में ये इमेज आती है, दैट इज कैसल्स नेकलेस डर्मेटाइटिस. दिस इज पेलाग्रा. So 34-year-old male was on maize as a staple diet. Maize, the niacin, it is present in the bound form, so it is unavailable for absorption. For prolonged period, he presented with diarrhea, lesion over the neck as shown in the image below. The condition occurs due to deficiency of niacin. It is due to deficiency of niacin. नियासिन इज मतलब कंप्लीट विटामिन नहीं है, क्योंकि कुछ पार्ट जो है वो हमारे बॉडी में ट्रिप्टोफैन से बनता है नियासिन का. सो दैट इज फ्रॉम द ट्रिप्टोफैन. सो द प्रोसेस और द inhibitors of the tryptophan metabolism also there is NAD deficiency, and that gives the pellagra like symptoms. A 35-year-old farmer on maize diet with diarrhea, dementia, and dermatitis. Which vitamin is deficient in this patient? So it is maize, diarrhea, dementia, and dermatitis. Classical 3Ds. Though it is niacin deficiency. See, this is Castle's necklace dermatitis. तो अभी ये मैं पढ़ूंगा नहीं. So it is related with the tryptophan. These complaints may be caused by deficiency in which of the following amino acids? So it is answer is tryptophan. Now the sulfur containing amino acid cysteine methionine. Sulfur containing amino acid metabolism, and this is subluxation of lens, ectopia lentis. This is a classical feature of homocystinuria. Homocystinuria, ectopia lensil, inferonasal subluxation of the lens occurs in the homocystinuria because this elevated homocysteine level, it interferes with the it causes the free radical formation. It interferes with the collagen cross links, and it interferes with the zonular fibers of the lens, and it causes the ectopia lentis, that is subluxation of the lens. So sulfur containing amino acid मतलब कौन से amino acids? Cysteine, methionine. So how it is metabolized? So cysteine and methionine. So they are incorporated into a protein. So they are incorporated into a protein. These are sulfur containing amino acids. Cysteine, methionine, sulfur containing amino acids. Then cysteine, it is polar. This methionine, it is non-polar. Methionine is the non-polar. It contains sulfhydryl group. It contains thioether linkage. Sulfhydryl group, it contains thioether linkage. So this is the NH2CH COOH, it is CH2 sulfhydryl group. So this is cysteine, sulfhydryl group. It is cysteine. In methionine, it is the thioether linkage. Sulfur is not free there, so it is in the thioether linkage. Cysteine and methionine. Now in the metabolism, they are glucogenic amino acid. These both are glucogenic amino acid, cysteine and methionine. They will give you the glucose. Okay, and there are different products they are given by the cysteine and methionine. We'll discuss this along with the homocystinuria. See here, methionine in the metabolism. Methionine, it is converted into S-adenosyl methionine. So adenosyl group is transferred to the sulfur of methionine. From the ATP, the adenosyl group is transferred to the sulfur of methionine. In presence of methionine, methionine adenosyl transferase, it forms the S-adenosyl methionine. Here sulfur is in the sulfonium form, and S-adenosyl methionine form the S-adenosyl homocysteine. So there is transfer of methyl group, which is accepted by an acceptor, and it is converted into methylated products. So it is converted into methylated product. For example, norepinephrine, it is converted into epinephrine, or epinephrine is converted into metanephrine, methyl transferase reaction. And this S-adenosyl homocysteine, by the homocysteinase, it is converted into homocysteine. homocysteine, and this homocysteine is converted into methionine. So this is methionine adenosyl transferase. This is methyl transferase reaction. So this is homocysteinase, adenosyl homocysteinase. There is removal of adenosyl group, and this is the methionine synthase reaction. Methionine synthase reaction, where homocysteine is converted into methionine. For this reaction, methyl tetrahydrofolate is required. Methyl THF is required, which is converted into THF. Methyl THF is converted into THF, and this is B12 dependent reaction. This is B12 dependent reaction, and also folic acid dependent reaction. So this is transmethylation reaction here, and examples of transmethylation reaction: epinephrine into metanephrine, norepinephrine into epinephrine, N-acetyl serotonin into melatonin, gonadotropin into creatinine. The ethanolamine is converted into choline like this. These are the different transmethylation reaction with the help of methyl transferase reaction, where S-adenosyl methionine is the universal methyl group donor. So this is this is important. This is folate trap. In the B12 deficiency, there is also the functional folate deficiency, because there is no use of methyl tetrahydrofolate. So because of this reaction, these two are connected with each other, and in the deficiency of these both, there is megaloblastic anemia, because they are important for this tetrahydrofolate is important for the C2C8 of the purine synthesis. It is important for the thymidylate synthesis. So deficiency causes the megaloblastic anemia. In B12 deficiency, folate deficiency. This is folate trap reaction. If there is B12 deficiency, it causes the functional folate deficiency. So in methionine metabolism, first step is the transmethylation reaction, where methionine is converted into homocysteine. So methionine is converted into S-adenosyl methionine, which is converted into S-adenosyl homocysteine, which is converted into homocysteine. Now this homocysteine, it is converted, it is combined with the serine to form cystathionine, and this cystathionine is converted into cysteine plus homoserine. Cysteine plus homoserine. Now this is second step in the methionine metabolism, where homocysteine is converted into cysteine, and third step is the degradation of cysteine or specialized product given by the cysteine. So first step is the transmethylation reaction, second conversion of homocysteine to cysteine, and third step is the conversion of cysteine into the specialized product or utilization of the cysteine. So deficiency of this enzyme, so this is cystathionine beta synthase. Pyridoxal phosphate dependent reaction. If there is deficiency of this enzyme, it causes homocysteinuria. There is elevated level of homocysteine, there is elevated level of methionine, but there is decreased level of cysteine. So cysteine is given in the treatment. Also B12 and folic acid, they are somehow useful in this disorder. They are useful in this disorder. In some cases, variants of this homocysteinuria, they are useful. So we will give the cysteine in the treatment in the patient with homocysteinuria. So there is subluxation of lens, ectopia lentis. In the homocysteinuria, cyanide nitroprusside test will be positive. There is myopia in this disorder, and there is serious manifestations because of increased homocysteine level. Because increased homocysteine, it forms the free radicals, it thiolates the it causes the thiolation of the LDL particles, and it leads to the formation of oxidized LDL formation because of elevated homocysteine level. So oxidized LDL formation, it is important for the atherosclerosis and the coronary artery diseases. So there is risk of having the coronary artery diseases because of elevated homocysteine level. So homocysteinuria, the typical feature is the ectopia lentis, then the ectopia lentis, cardiovascular disorders because of elevated homocysteine level, myopia. There is myopia. There is it miss the classical picture. First MBBS को ectopia lentis या subluxation of lens, ये word रहेगा ही रहेगा. Okay. So this is all about the homocysteine urea. and the methionine metabolism. Methionine is converted into homocysteine, then homocysteine is converted into cysteine, and cysteine gives specialized products. So cysteine, it will give you the cotapaji. This is coenzyme A, taurine, taurine, the pyruvate, which will give you glucose, and glutathione, glutathione. So cysteine, so it will give you coenzyme A, taurine. It will give you phosphoadenosine phosphosulfate, phosphoadenosine phosphosulfate. It will give you pyruvate, which is converted into glucose. So coenzyme A, phosphoadenosine phosphosulfate, taurine, pyruvate, and glutathione, they are produced from the cysteine. So this is all about the methionine and cysteine metabolism. Methionine and cysteine metabolism. Now we'll discuss the glycine in short. So there are four important reactions in which glycine is synthesized in our body. Four important reactions. First one is the conversion of serine into glycine by the serine hydroxymethyl transferase reaction. Serine is converted into glycine. Second one is the threonine is converted into glycine. Threonine is converted into glycine. This is optically inactive amino acid. Smallest amino acid, contributes 30% to the collagen. It is important in the collagen structure, and as it is a smaller molecule, it can occupy in the turns in the collagen molecule. So serine is converted into glycine. Threonine is, by the threonine aldolase reaction, it is converted into glycine, and the glycine cleavage system. So NH3CO2 and N5N10 methylene tetrahydrofolate by the glycine cleavage system, by the glycine synthase. Glycine synthase, it will form the glycine. It will form the glycine synthase system, and glyoxylate. Glyoxylate will give you glycine by the glyoxylate aminotransferase. It will form the glyoxylate aminotransferase. So these are the four reactions which are important. Transferase, four reactions which are important in the glycine synthesis. Serine hydroxymethyl transferase, serine is converted into glycine. This is reversible reaction. So second, threonine aldolase, threonine is converted into glycine. Then glycine cleavage system, where NH3CO2 and N5N10 methylene tetrahydrofolate will give you glycine, and from the glyoxylate aminotransferase, it will give you glycine. Now how degradation of glycine occurs? So glycine, by the glycine cleavage system, this is degraded. Just wait. Now the degradation of glycine. How glycine is degraded? So glycine, it is converted into serine. So this is reversible reaction. This was reversible reaction. Second, glycine, by the glycine oxidase, it is converted into glyoxylate. And glyoxylate, it is converted into formate. It is converted into oxalate. It is converted into oxalate, formate, oxalate, and one more is there, malate. So it enters the TCA cycle. This oxalate, it is excreted in the urine, and formate enters the 1-carbon metabolism. So glyoxylate, by the glycine oxidase. So glycine oxidase, it is converted into glyoxylate, and it is converted into oxalate and excreted in the urine. So glycine is converted into serine, glycine is converted into glyoxylate. So via serine, it will give you glucose. And the glycine, by the glycine cleavage system, it is converted into CO2, NH3, and N5N10 methylene tetrahydrofolate. So this is how glycine is degraded in our body. There are various specialized product given by the glycine. So glycine, it is incorporated into a collagen structure. 30% is the glycine, that is glycine XY repeat in the collagen structure. It is important for the synthesis of collagen. It is glucogenic amino acid, it will give you glucose, and the specialized product, it will give you heme. The first step, of heme synthesis, glycine is important. Second, it is important in the glutathione structure. That is gamma glutamyl cysteine glycine. Then it is important in the conjugation reaction. Then it is important in the C4, C5 and N7 of the purine ring. Then glycine, it is important. So heme, glutathione, conjugation, then formation of creatine and creatine phosphate. Creatine phosphate, it is important. So these are the important products given by the glycine: heme synthesis, glutathione synthesis, conjugation reaction, purine ring formation, and creatine phosphate synthesis. Glycine is important, and in collagen it is 30% glycine. So this is all about the glycine metabolism. In the branch chain amino acid, just 3 steps are there. Just remember 3 steps. So valine, isoleucine and leucine, they will form the alpha keto acids. And this alpha keto acid dehydrogenase, they will form acyl coenzyme A. Respective acyl coenzyme A they will form. And see, all of you know that leucine will give you acetoacetate. So it will give you fat. Leucine will give you fat. Isoleucine will give you both glucose and fat, and the valine will give you only glucose. So 2 steps are important here. One is the transamination. By the transamination, this valine, leucine and isoleucine, they are converted into respective alpha keto acid, and branch chain keto acid dehydrogenase, it converts the keto acid into acyl coenzyme A, and they will give this respective products: valine, isoleucine, leucine. Valine is glucogenic, isoleucine is both glucogenic and ketogenic. So it will give you the succinyl coenzyme A, and it will give you the acetoacetate, and leucine will give the acetyl coenzyme A or acetoacetate, and that will give you a fat. So if there is defect in this branch chain keto acid dehydrogenase, there is increased level of alpha keto acid and these amino acids in the urine, that causes the maple syrup urine disease. So maple syrup urine disease, it is due to branch chain alpha keto acid dehydrogenase complex defect. So it causes MSUD. So there is excretion of alpha keto acids in the urine, which give smell like burnt sugar. So it is lethal condition. So mental retardation is there, neurological manifestations will be there. There is hypotonia, muscle tone is altered in this disease, and there is increased excretion of the keto acids. So it can cause the metabolic acidosis, because bicarbonate is utilized for buffering these acids and gives the maple syrup or burnt sugar like smell. So this is days old infant brought to hospital with poor feeding, convulsion and vomiting. Urine had smell of burnt sugar. Biochemical analysis suggestive of increased branch chain amino acid and their keto acids in the urine, and ABG shows low bicarbonate level. So what is your diagnosis here? It is maple syrup urine disease. So neurological manifestations, burnt sugar, vomiting is there, and increased alpha keto acids in the urine. Because of defect in the branch chain keto acid dehydrogenase, it causes the maple syrup urine disease. So this is all about the amino acid chemistry, amino acid metabolism, प्रोटीन केमिस्ट्री, प्रोटीन मेटाबॉलिज्म, इंपॉर्टेंट टॉपिक्स. एक छोटा मेरा बायोजेनिक अमाइंस. So this is by the decarboxylation, pyridoxal phosphate dependent reaction. There is synthesis of tyramine, there is synthesis of dopamine, there is synthesis of the taurine, there is synthesis of the GABA from the glutamate. So various neurotransmitters, they are produced by the decarboxylation reaction pyridoxal phosphate. When the amino groups are multiple amino groups, it is known as the polyamines. So it includes putrescine, spermidine, spermine. These are the examples. They are useful in the growth and differentiation, and they are marker for the different cancers. So download the Meded app and explore the FARRE series. So notes will be available on the app. So this is all about the amino acid chemistry and metabolism. Thank you all of you. One carbon metabolism important है. Just क्या बोलते हैं उसको, फॉर्मेल टीएचएफ से क्या बनता है, मिथिल टीएचएफ से क्या बनता है, मिथाइलिन टीएचएफ से क्या बनता है, और मिथेनिल टीएचएफ से क्या बनता है. ये आप कर लो एक के बाद एक, तो वन कार्बन मेटाबॉलिज्म हो जाएगा आपको आपका. ओके, so this is all about the amino acid chemistry and metabolism. Thank you all of you. Thank you for patient listening. Thank you Sneha ji, thank you Pratham, thank you Vinay, thank you Yogesh, thank you all of you.